Hello, and welcome. I'm glad you're here. Tonight we're going to explore something extraordinary, something that's been with you since before your first breath, working quietly in the background of every moment you've ever experienced. We're talking about your nervous system, that vast, intricate network of electrical whispers and chemical conversations that makes you, you. It's the reason you can feel the weight of your blanket right now, the reason you can understand these words, the reason you know where your hand is even with your eyes closed. It's been orchestrating your heartbeat since before you were born, and it will keep doing so, quietly, faithfully, through every moment of rest and waking. Before we begin our journey tonight, I'm curious. How was your day? Whatever it was, challenging, peaceful, ordinary, overwhelming, I'd love to hear about it in the comments below. And if you find these quiet explorations helpful, if they bring you a bit of peace at the end of your day, please consider subscribing. It helps me know these late-night conversations matter to someone out there in the darkness. Now, let's begin. Your nervous system contains approximately 86 billion neurons, each one a tiny living computer processing electrical signals. Let's start with something almost impossible to comprehend. Right now, as you lie there reading or listening to these words, there are approximately 86 billion neurons inside your skull. 86 billion! That's not a metaphor or an exaggeration. It's a careful count, arrived at through painstaking scientific work. Each one of these neurons is a living cell, a tiny biological computer that processes information through electrical signals. They're not passive wires, not simple on-off switches. Each neuron is making decisions, integrating information from thousands of other neurons, deciding whether to fire or stay silent. And they're doing this constantly, every moment, even now as you begin to settle in for sleep. If you wanted to count them, 1 neuron per second, no breaks for sleep or food, it would take you nearly 3,000 years to count them all. By the time you finished, human civilization would have risen and fallen several times over. And yet, here they all are, packed into the space of your skull, working together to create your thoughts, your memories, your sense of being someone at all. It's a bit like having a city more populous than Earth itself tucked quietly inside your head. Neurons are the oldest cells in your body. Most were born before you were and will never be replaced. Here's something that might make you pause. The neurons reading these words right now, the ones processing the meaning of this sentence, converting symbols into understanding, they've been with you since before your first memory. Most of them were born before you were, formed in the quiet darkness before birth, and they will never be replaced. Unlike your skin cells, which renew every few weeks, or your red blood cells, which cycle through every few months, most of your neurons are as old as you are. They were there for your first breath, your first word, your first heartbreak, your first joy. They've accumulated every experience you've ever had, storing it somehow in the pattern of their connections. It's a strange kind of permanence in a body that's otherwise in constant flux. The atoms in your neurons get replaced, carbon swapped for carbon, oxygen for oxygen, but the cells themselves persist. In a very real sense, you are your neurons. They're the continuity that threads through all the changes, the stable core around which everything else revolves. And tonight, as you drift towards sleep, these ancient cells will shift into a different mode, consolidating memories, clearing away metabolic debris, preparing for another day of being you. A single neuron can connect to up to 10,000 other neurons, creating a network more complex than the entire internet. Imagine a single neuron, just one cell among those 86 billion. Now imagine that this one cell can reach out and touch up to 10,000 other neurons, forming connections, passing messages, participating in vast electrical conversations. And now remember, every single one of those 86 billion neurons is doing the same thing. The mathematics become almost absurd. Your brain contains more possible connections than there are stars in the Milky Way galaxy. More pathways than all the roads, fiber optic cables and shipping lanes on Earth combined. The entire internet, every website, every server, every connection is vastly simpler than the network inside your skull. And unlike the internet, which was designed by engineers with blueprints and plans, your neural network assembled itself. It started with a few cells and grew through a process of organized chaos, neurons reaching out, finding partners, strengthening useful connections, pruning away the ones that don't serve. It's still doing that now, actually. Even as an adult, even as you read this, connections are being made and unmade, the network constantly reshaping itself based on your experiences. You are, in the most literal sense, rewiring yourself with every thought. The fastest nerve signals in your body travel at 268 miles per hour, faster than a Formula One race car. Your nervous system speaks in lightning. The fastest signals, the ones that control your muscles, that let you catch a falling object or pull your hand back from danger, travel at approximately 268 miles per hour. That's faster than any car on any highway, faster than most small aircraft. These signals can travel from your toe to your brain in about 20 milliseconds, a span of time so brief you can't even perceive it. But here's what makes it even more interesting. Not all your nerve signals travel at the same speed. Some crawl along at barely 2 miles per hour, slower than you walk. These slow signals carry information about dull, aching pain, about gradual changes in temperature, about the vague sense that something might not be quite right. Your body has, in essence, multiple communication networks running at different speeds. The fast network handles emergencies, quick reflexes, rapid movements, immediate threats. The slow network handles background information, ambient sensations, things that don't require instant response. It's a bit like having both a phone call and a letter system working simultaneously, each one suited to different kinds of messages. And all of this is happening right now, beneath your awareness, as your nervous system monitors and maintains your existence. Your brain generates enough electricity to power a small LED light bulb. Every thought you have is electrical, not metaphorically, literally. Your neurons generate electrical signals, tiny voltages that pulse and flow through your brain's circuitry. And if you could somehow harvest all that electricity, all those millions of tiny sparks happening simultaneously, you could power a small LED light bulb, about 10 to 20 watts of continuous electrical power generated by thought itself. It's a strange image, isn't it? The light by which you're perhaps reading this, or the glow of your screen, your brain generates about that much power constantly, not through combustion or mechanical motion, but through the coordinated firing of billions of neurons, each one maintaining a tiny electrical gradient across its membrane, each one occasionally unleashing that stored energy in a brief spike. And unlike a light bulb which converts most of its energy into heat, your brain is remarkably efficient. Those 10 to 20 watts support not just electrical activity, but all the chemical processes of thinking, remembering, imagining, dreaming. Every memory you've ever formed, every word you've ever spoken, every plan you've ever made, all of it powered by less electricity than it takes to light a reading lamp. In the quiet of this moment, in the dimness of evening, there's a kind of poetry in that. Consciousness itself running on the power of a gentle glow. The word neuron comes from the Greek word for sinew, or cord, because early anatomists thought nerves were like strings. There's something endearing about the history of how we've understood the nervous system. The word neuron comes from ancient Greek, specifically from the word for sinew, or cord. When early anatomists first dissected bodies and found these pale, stringy structures running through flesh, they thought they were looking at some kind of biological rope, cords that connected parts of the body to the brain, like puppet strings or the rigging of a ship. It's not a terrible analogy, really. Nerves do look like strings when you encounter them in dissection. They're tough, fibrous, organized into bundles, and in a sense, they do transmit signals from one place to another, carrying information from distant outposts back to headquarters. But the early anatomists had no idea what they were really looking at. It wasn't until the 1830s that scientists realized these cords were actually made of individual cells, living units that could generate and transmit electrical signals. And it took even longer to understand that each of these cells was mind-bogglingly complex, a complete biological system in its own right. The word neuron has stuck, though, carrying with it a reminder of how recently we've understood what we really are. Just 200 years ago, we thought our thoughts travelled along strings. Your spinal cord stops growing when you're about 4 years old, even though you keep getting taller. Here's something curious about development. Your spinal cord, that crucial highway of information running down your back, stops growing when you're about 4 years old. 4. After that, even though you keep getting taller, perhaps doubling or tripling in height by adulthood, your spinal cord stays essentially the same length. So what happens? Your vertebrae, the bones that protect your spinal cord, keep growing. Your back gets longer, but the spinal cord inside doesn't keep pace. This creates increasing space between where nerve roots exit the spinal cord and where they pass through gaps in the vertebrae. In adults, these nerve roots hang down like the roots of a plant, a structure anatomists call the cauda equina, the horse's tail, because that's exactly what it looks like. It's a reminder that growth isn't uniform, that different parts of you developed on different schedules. Your brain grew rapidly in your first few years. Your spinal cord achieved its adult length while you were still in preschool. Your bones kept extending into your teens or early 20s. You are, in a sense, an assembly of parts that matured at different rates. You're finally coming together into the person you are now, lying here in the quiet, all those different developmental timelines having converged into this single moment. There are more neurons in your gut than in your spinal cord, about 500 million of them. Your gut has a brain, not metaphorically. It has an actual nervous system, complete with about 500 million neurons. That's more neurons than you have in your entire spinal cord. This network, called the enteric nervous system, aligns your digestive tract from esophagus to rectum, and it can operate entirely independently of your brain. This second brain makes decisions about digestion, controls the movement of your intestines, regulates the release of enzymes and hormones, and even influences your immune system. It does all of this without consulting the brain in your skull. In fact, more signals travel from your gut to your brain than from your brain to your gut. Your digestive system is, in a sense, talking to your brain more than your brain is talking to it. This is why gut feelings are more real than you might think. That sensation of butterflies when you're nervous, that sinking feeling when something's wrong, that sense of calm after a good meal. These aren't just metaphors. They're your gut's nervous system communicating with your brain, participating in your emotional life in ways we're only beginning to understand. As you lie here tonight, your gut brain is working away quietly, managing the complex process of digestion, sending status updates to your conscious mind, being part of you in ways that happen mostly beneath awareness. When you learn something new, your brain physically changes shape as neurons form new connections. Right now, as you take in these words and ideas, your brain is physically changing. Not in some abstract or metaphorical sense, it's literally reshaping itself. Neurons are reaching out, forming new connections with their neighbors. Synapses are strengthening or weakening. The physical structure of your brain is different now than it was when you started reading this sentence. This process, called neuroplasticity, means that learning isn't just about storing information, it's about reconstruction, about physically altering the architecture of your nervous system. When you practice a skill, certain neural pathways get reinforced, growing stronger and more efficient. When you remember an event, you're not accessing a file stored somewhere. You're recreating a pattern of neural firing, and each time you do it, you slightly change that pattern. This is happening all the time, constantly, even in sleep. Tonight, as you drift off, your brain will replay the day's experiences, strengthening some memories, letting others fade, literally sculpting itself based on what you've lived through. You wake up tomorrow in a brain that's physically different from the one you had today. In the gentlest, most gradual way imaginable, you're always becoming someone slightly new. Your nervous system processes 11 million bits of information per second, but you're only consciously aware of about 40 bits. Your nervous system is processing an overwhelming flood of information right now, about 11 million bits per second, flowing in from your senses, your internal organs, your muscles and joints. Your eyes alone send roughly 10 million bits per second to your brain. Your skin, your ears, your sense of balance, all of them contributing to this massive stream of data. But here's the remarkable thing. Of those 11 million bits per second, you're consciously aware of maybe 40 bits out of 11 million. Your brain is constantly making decisions about what's important enough to bring to your attention and what to handle quietly in the background. Most of your existence happens beneath consciousness. Your nervous system managing your heartbeat, your breathing, your digestion, your posture, the constant fine-tuning of muscle tension, the monitoring of thousands of internal variables. Right now, your conscious mind might be focused on these words, but your nervous system is simultaneously tracking the temperature of the room, the pressure of your body against the bed, the ambient sounds, the position of your limbs, the state of your digestion, and countless other factors. It's making judgments every millisecond about what deserves your attention, and mercifully, it decides that you don't need to know about most of it. You can rest, attend to just a few things, let the vast machinery of your nervous system handle the rest in quiet competence. Phantom limb sensations occur because your brain maintains a complete map of your body, even when parts are missing. Deep in your brain, there's a map of your body. Not a metaphorical map, an actual physical representation where specific regions of your cortex correspond to specific parts of your body. There's a patch of neurons for your left thumb, another for your right foot, another for your lips. This map is so detailed, so precise, that neuroscientists can predict exactly which part of your brain will activate when you move any particular body part. But here's what's haunting and beautiful about this map. It's remarkably stable. It forms early in development and persists throughout your life. So stable, in fact, that even when a limb is lost, through amputation or injury, the map remains. The neurons that once received signals from that hand or foot are still there, still organized in the same pattern, still expecting input that will never come. This is why phantom limbs feel so real. People can feel an amputated hand clench, feel toes that no longer exist curl and stretch. The representation of the body and the brain is so deeply embedded, so fundamental to how the nervous system organizes itself, that the brain sometimes refuses to update the map. It's as if your nervous system is saying, I know what a complete body feels like, and this is what it should be. There's something profound in that stubbornness. The brain holding on to a version of you that no longer physically exists, maintaining a memory written in neural architecture itself. Your neurons communicate using both electrical signals and chemical messengers called neurotransmitters. Communication in the nervous system is a hybrid affair, part electrical, part chemical, with both languages working together in an intricate dance. When a neuron fires, it sends an electrical signal racing down its length at incredible speeds. But when that signal reaches the end of the neuron, something remarkable happens. It can't jump directly to the next neuron. There's a gap, a tiny space called a synapse, just 20 to 40 nanometers wide, 5,000 times thinner than a human hair. So the electrical signal triggers the release of chemicals, neurotransmitters, that float across this gap and bind to receptors on the next neuron. It's like a conversation conducted in two languages simultaneously: electricity for speed and long-distance transmission, chemistry for precision and control. The chemical step allows for modulation, for fine-tuning, for complexity that pure electrical signals couldn't achieve. Think of it like this: electrical signals are the words, but neurotransmitters are the tone of voice, the facial expressions, the emotional context that gives those words meaning. A neuron can say different things depending on which neurotransmitter it releases, and the receiving neuron can interpret the same neurotransmitter differently depending on which receptors it expresses. It's a communication system of extraordinary subtlety, and it's happening trillions of times per second throughout your nervous system right now. As you read these words and begin to feel the gentle pull of sleep, the myelin sheath that insulates nerve fibers is white, which is why brain tissue has distinct white matter and grey matter. If you could slice open a brain and look at it in cross-section, which mercifully you don't need to do, you'd see distinct regions of different colours. Some parts look greyish pink, others appear almost white. This isn't just variation in tissue density. It's a fundamental architectural distinction that tells you something profound about how the brain is organised. The grey matter is where neuron cell bodies cluster together, the parts that do the actual computational work, the processing, the decision-making. The white matter is mostly the axons, the long fibers that connect distant brain regions, wrapped in a fatty insulation called myelin. And myelin, it turns out, is white, or rather, whitish. It's this insulation that gives white matter its characteristic appearance. Grey matter thinks. White matter connects the thinkers. It's a bit like the difference between cities and highways. The grey matter is where the activity happens, where neurons cluster and interact, and the white matter is the infrastructure that lets different regions communicate. Your brain is organized into these thinking centers connected by high-speed communication lines. Here's all color-coded by the basic biology of insulation. And tonight, as you drift towards sleep, both systems remain active. Grey matter processing the day's memories, white matter ensuring all the distant parts of your brain stay synchronized in their slow rhythms of rest. Your brain uses 20% of your body's oxygen, despite being only 2% of your body weight. Your brain is expensive, not in monetary terms, but in metabolic ones. Despite making up only about 2% of your body weight, roughly 3 pounds in an average adult, your brain consumes approximately 20% of all the oxygen you breathe. 1/5 of every breath you take is dedicated to keeping your neurons functioning. This is because thinking is metabolically intense. Every electrical signal, every neurotransmitter released and recycled, every ion pumped across a membrane, all of it requires energy. Your neurons are constantly working, maintaining their electrical gradients, ready to fire at a moment's notice. Even at rest, even in sleep, the brain remains one of the most active organs in your body. This means that a hard day of mental work can actually burn a significant number of calories. Your brain weighs about as much as a laptop computer, but it consumes as much energy as your leg muscles during a moderate walk. Problem solving, learning, concentrating. These aren't just abstract mental activities. They're physical processes that consume real resources, that require fuel and oxygen, that can leave you genuinely tired. So if you feel exhausted after a day of thinking, you're not being dramatic. Your brain has been running at high speed, burning through oxygen and glucose, working as hard as any muscle in your body. There are over 100 different types of neurons, each specialized for different tasks. When we talk about neurons, it's easy to imagine them as more or less identical, basic units all doing the same job. But the reality is far more interesting. There are over 100 different types of neurons in your nervous system, each one specialized for particular tasks, each one unique in its shape, its connections, and its function. Some neurons respond only to vertical lines in your visual field. Others respond only to horizontal lines. Some fire only when you see something moving left, others only for rightward motion. There are neurons that detect sweetness on your tongue, others that detect bitterness, sourness, saltiness, umami, each taste having its own dedicated detector cells. There are neurons specialized for detecting pain, others for gentle touch, others for vibration, others for steady pressure. This specificity extends throughout your nervous system. Your brain doesn't have general-purpose neurons that do a bit of everything. It has specialists, neurons that have evolved and developed to do one thing exceptionally well. It's like an orchestra where some musicians only play high notes, others only low ones, some only play during crescendos, others only during quiet passages, and somehow, from all these specialists doing their narrow jobs, emerges the full symphony of experience, the integrated, unified sense that you are one person, experiencing one coherent world. The gap between neurons, called a synapse, is only about 20 to 40 nanometers wide, 5,000 times thinner than a human hair. Every thought you've ever had, every memory you've ever formed, every decision you've ever made has required signals to cross a gap almost impossibly small. The synapse, the space between one neuron and the next, is typically about 20 to 40 nanometers wide. To put that in perspective, that's about 5,000 times thinner than a human hair. You could stack about 2,500 synapses in the width of a single bacterium. Yet this tiny space is where all the complexity happens. This is where electrical signals get translated into chemical ones, where neurotransmitters are released and received, where the strength of connections can be adjusted, where learning physically happens. Every time you remember something, you're strengthening synapses. Every time you forget, synapses are weakening or being eliminated entirely. And the remarkable thing is that signals cross this gap in less than a millisecond. Chemical messengers are released, drift across this nanoscale canyon, bind to receptors, trigger electrical changes in the receiving neuron, all faster than you can blink. Trillions of these crossings are happening right now throughout your brain, messages passing through these impossibly small spaces, creating the continuous flow of thought and sensation that we call consciousness. The gap is so small you couldn't see it even with a powerful microscope, and yet every aspect of your mental life depends on what happens there. Your nervous system begins forming just 16 days after conception, before most people even know they're pregnant. You began as a nervous system, not quite, but almost. Just 16 days after conception, before the embryo is even a millimeter long, before there's anything that looks remotely human. the first neural tissue begins to form. It starts as a flat sheet of cells called the neural plate, which then rolls up into a tube, the neural tube, that will eventually become your brain and spinal cord. This makes the nervous system the first organ system to develop, and there's a good reason for that. Nothing else can form without instructions from the nervous system. Your heart needs neural signals to beat in the right rhythm. Your muscles need nerves to tell them when to contract. Your digestive system needs neural networks to coordinate its movements. Everything depends on the nervous system, so the nervous system has to come first. At 16 days, the embryo doesn't have eyes yet, or ears, or limbs, or a heart. But it has the beginnings of a nervous system. In a sense, you were a nervous system before you were anything else. A small network of cells that would eventually grow into the intricate, beautiful system that's reading these words right now. That original neural tube formed before your mother might have even known you existed, and quietly assembling the foundation of everything you would become. Neurons can fire up to 200 times per second, creating rhythmic patterns that researchers can actually hear when converted to sound. Your thoughts have a rhythm. Neurons don't fire randomly or continuously. They fire in pulses, discrete electrical spikes that happen at varying rates. Some neurons fire slowly, maybe once or twice per second. Others fire rapidly, up to 200 times per second during intense activity. And when large groups of neurons fire together, they create synchronized rhythms, waves of electrical activity that sweep across the brain. Researchers have learned to listen to these rhythms by converting the electrical signals into sound. When they do, they hear something remarkable. Your brain has a soundtrack. Different states of consciousness have different sounds. Deep sleep has slow, regular waves. Active thinking has rapid, complex patterns. Seizures have distinctive, alarming rhythms. The nervous system is, in a literal sense, making music. Electrical music that normally exists beyond human hearing, but that can be translated into sound. These rhythms aren't just noise. They seem to be how different brain regions coordinate their activity, how they synchronize to work together. When you focus attention on something, certain brain regions start firing in sync, their rhythms aligning like instruments in an orchestra finding the same tempo. Right now, as you settle towards sleep, your brain rhythms are slowing, deepening, shifting from the quick, irregular patterns of wakefulness to the slow, regular waves that characterize rest. You can't hear it, but your nervous system is playing its nightly lullaby, conducting itself gently towards sleep. The human brain has the same basic structure as a mouse brain, a whale brain, and even a dolphin brain. There's a blueprint for the mammalian brain, and evolution has been remarkably conservative with it. Whether you're a mouse weighing 20 grams or a blue whale weighing 200 tons, the basic architecture is the same. A cerebral cortex folded around deeper structures, a cerebellum in the back for motor control, a brainstem connecting to the spinal cord, a limbic system for emotions and memory. The same basic parts, arranged in the same basic way. What differs is mostly scale and specialization. A whale brain is larger, with more neurons dedicated to processing sound, crucial for an animal that navigates by echolocation in the ocean's darkness. A mouse brain is smaller, with proportionally more neurons dedicated to whisker sensation and smell. Human brains have an extraordinarily large prefrontal cortex relative to our body size, giving us our distinctive capacity for planning, abstraction, and self-control. But the fundamental design, the way neurons organize into layers, the way different regions connect, the basic neurochemistry, all of that is shared. You and a mouse are using variations of the same neural software, running on similar hardware. Evolution found a design that works and has been refining it, scaling it, specializing it for different niches for over 100 million years. In a sense, your brain is ancient, built on a plan that predates humanity by countless generations. Your sense of touch is actually thousands of tiny nervous system sensors in your skin, each specialized for different sensations. What you call touch is actually many different senses working together, each with its own specialized receptors embedded in your skin. Some receptors detect steady pressure. They're responsible for feeling the weight of your blanket or the contact of your body against the mattress. Others detect vibration. These let you feel the buzz of a phone or the texture of fabric as you run your fingers across it. Still others detect stretching of the skin, saw movement of hairs, or changes in temperature. Each type of receptor is a miniature biological machine, exquisitely sensitive to its particular stimulus. The receptors that detect vibration, called Pacinian corpuscles, can sense displacements smaller than the wavelength of visible light. The receptors for pain, called nociceptors, can detect tissue damage at the molecular level, warning you of harm before significant damage occurs. And all of these receptors, thousands of them across your skin, are sending their individual reports to your brain constantly, every moment. Your nervous system integrates all these separate signals into the unified sensation you experience as touch. Right now, you're probably aware of only a few touch sensations, perhaps the pressure of your head on the pillow or the temperature of the air on your face. But your skin is sending thousands of reports every second, most of which your brain quietly processes without bothering your consciousness. It's only when something changes, when something demands attention, that these background sensations rise into awareness. Anesthesia works by disrupting communication between neurons, but scientists still don't fully understand how. Here's something unsettling and fascinating. We use anesthesia millions of times every day across the world. We trust it with our lives during surgery, yet we understand that it works reliably and safely. And yet we don't fully understand how it works. We know it disrupts communication between neurons, that it somehow prevents the brain from processing pain and maintaining consciousness, but the precise mechanisms remain mysterious. Different anesthetics seem to work in different ways. Some interact with neurotransmitter receptors, enhancing inhibitory signals that quiet neural activity. Others seem to affect the physical properties of neuron membranes, making them less excitable. Some might disrupt the coordination between different brain regions, preventing the synchronized activity that seems necessary for consciousness. But there's no single, unified theory that explains how these drugs can so reliably turn consciousness off, and then, just as reliably, allow it to return. It's one of medicine's most important tools and one of its deepest mysteries. We can make you unconscious with remarkable precision, keep you in that state for hours, wake you up with no memory of the time that passed, and we're not entirely sure how we're doing it. The nervous system guards its secrets well. Even our most sophisticated interventions sometimes work through mechanisms we're still trying to understand. As you drift towards sleep tonight, natural sleep, not an aesthetic unconsciousness, your brain is transitioning through states that are, in their own way, equally mysterious. Your autonomic nervous system controls your heartbeat, breathing, and digestion without you thinking about it. Right now, as you read this, your heart is beating, about 60 to 100 times per minute, steadily, reliably, without any conscious effort on your part. Your lungs are expanding and contracting, drawing in air, extracting oxygen, expelling carbon dioxide. Your digestive system is working on whatever you ate today, moving food through your intestines with coordinated muscular contractions. Your pupils are adjusting to the light. Your body temperature is being regulated. Your blood pressure is being maintained. None of this requires your attention. You don't have to remember to beat your heart or remind yourself to breathe. Your autonomic nervous system, the automatic unconscious part of your nervous system, handles all of it. It's been doing so since before you were born, and it will continue until the moment you die, a faithful servant that never sleeps, never takes a break, never asks for recognition. You're only alive right now because your nervous system is managing thousands of tasks you're not even aware of. It's monitoring your blood oxygen levels, adjusting your breathing rate accordingly. It's tracking your blood glucose, signaling when you need to eat. It's coordinating the muscles in your digestive tract, the contractions of your bladder, the dilation of your blood vessels. All of this happens in the background beneath consciousness, managed by neural circuits that work tirelessly to keep you alive. You're free to think about other things, to read, to dream, to wonder, precisely because your autonomic nervous system is handling the essential business of existence. Mirror neurons fire both when you perform an action and when you watch someone else do it. In the 1990s, the researchers studying monkey brains made an accidental discovery that would transform our understanding of empathy and social connection. They found neurons that fired not only when a monkey reached for food, but also when the monkey watched a researcher reach for food. The same neurons activated for both actions, performing and observing. They called these mirror neurons. Humans have them too. When you watch someone pick up a cup, neurons in your motor cortex activate as if you were picking up the cup yourself. When you see someone smile, your brain simulates that smile internally. When you watch someone stub their toe, neurons fire in your pain centers, not as intensely as if you'd stubbed your own toe, but enough that you genuinely feel a shadow of their pain. This is why watching someone get hurt makes you wince, why yawns are contagious, why watching a skilled athlete can help you learn the movement yourself. Your brain doesn't just observe other people, it simulates their experiences internally, creating a kind of neural resonance. You understand others, at least in part, by briefly becoming them in your imagination. It's a profound form of connection, written into the architecture of the nervous system itself. We are, quite literally, built to share in each other's experiences. The blood-brain barrier is a special filtering system that protects your nervous system from toxins. Your brain is precious and vulnerable, and your body knows it. Surrounding the blood vessels in your brain is a specialized filtering system called the blood-brain barrier, a selective boundary that carefully controls what can pass from your bloodstream into your brain tissue. It's made of tightly packed cells that block most molecules from crossing, allowing only specific substances through. Oxygen and glucose, which your brain needs desperately, can cross freely. But most drugs, toxins, and even beneficial medications are blocked. This barrier is so selective, so strict, that it presents one of the biggest challenges in neurology and psychiatry. A medication that could help a brain disease might be completely ineffective simply because it can't cross this barrier. Brain tumors are particularly difficult to treat because chemotherapy drugs often can't reach them. But this strict security makes sense. Your brain cells are irreplaceable, unlike your liver or skin, which can regenerate damaged tissue. Most brain damage is permanent, so your body maintains this careful barrier, inspecting everything that tries to enter, allowing through only what the brain needs and blocking nearly everything else. It's working right now, standing guard while you rest, ensuring that only the right molecules reach the delicate neural tissue that makes you who you are. Your cerebellum, which coordinates movement, contains more neurons than the rest of your brain combined. There's a structure at the back of your brain, tucked beneath the larger cerebral hemispheres, that looks almost like a separate, smaller brain. It's called the cerebellum, the little brain, and despite being only about 10% of your brain's volume, it contains more than half of all your neurons. More than 50 billion neurons packed into this relatively small structure, more than all the rest of your brain combined. The cerebellum's job is to coordinate movement, to make your actions smooth and precise. When you reach for a glass of water, your cerebral cortex initiates the movement, but your cerebellum constantly adjusts it in real time, coordinating dozens of muscles, compensating for the weight of your arm, predicting where the glass will be by the time your hand arrives. Walking, which feels effortless and automatic, requires incredibly complex calculations that your cerebellum performs continuously. This is why the cerebellum needs so many neurons. As a movement is computationally expensive, balancing on two legs while walking, adjusting your posture constantly to maintain equilibrium, coordinating eyes and hands to catch a ball, these seemingly simple actions require more processing power than solving complex math problems. Your cerebellum is working right now, even as you lie still, making tiny adjustments to your muscle tone, maintaining your posture, preparing for movement even in stillness. It's a computational powerhouse, quietly managing the physics of your existence. Nerve regeneration in adults is extremely slow, about 1 millimeter per day at best. Your neurons have a remarkable ability. Unlike most brain cells, peripheral nerves can regenerate after injury. If you cut a nerve in your arm or leg, the axon can slowly grow back, reconnecting with its target. But the process is excruciatingly slow, about 1 millimeter per day under the best conditions, sometimes slower, and this means that a nerve injury in your fingertip might heal in a few weeks, but a nerve injury near your shoulder trying to reach that same fingertip could take more than a year to fully regenerate, and that's assuming everything goes perfectly, that the regenerating axon finds the right path, reconnects with the right target, doesn't get tangled or misdirected. During this time, the part of your body served by that nerve remains numb or paralyzed, waiting for the slow biological construction project to complete. Recovering from severe nerve damage requires patience measured in months or years. Your nervous system can heal itself, but it does so at its own pace, 1 careful millimeter at a time. It's a reminder that for all its speed and complexity, the nervous system is still made of living tissue, subject to the slow processes of biological growth and repair. Your brain's neurons are supported by even more glial cells, which were once thought to be just glue. For most of neuroscience's history, the supporting cells in your brain, called glial cells, were thought to be relatively boring. The name itself means glue in Greek, reflecting the belief that these cells just held neurons in place, providing structural support and nothing more. Neurons did the important work of thinking, and glia were just the scaffolding. We were wrong. Gloriously, completely wrong. Glia turn out to be active participants in nearly everything the brain does. Astrocytes, one type of glial cell, listen to neural conversations and can strengthen or weaken synapses, directly influencing learning and memory. Microglia act as immune cells, but also actively prune unnecessary synapses, sculpting neural networks. Oligodendrocytes create the myelin insulation that speeds up signal transmission. In fact, you have more glial cells than neurons. They outnumber neurons by about 10 to 1. Your brain is, in a very real sense, more glia than neuron. These cells are actively involved in thinking, learning, remembering, and feeling. What we once dismissed as mere support staff turn out to be essential partners in cognition. It's a humbling reminder that the brain's complexity exceeds what we can see, even when we look directly at it. The more we learn, the more we discover how little we truly understood. The vagus nerve, which connects your brain to your gut, heart, and lungs, is sometimes called the wandering nerve. There's a nerve that wanders through your body like an explorer, reaching from your brainstem all the way down to your abdomen, branching off to touch your heart, your lungs, your digestive tract, and numerous other organs along the way. It's called the vagus nerve, from the Latin word for wandering, and ancient anatomists who first traced its path were astonished by how far this single nerve travels. The vagus nerve is the main channel of your parasympathetic nervous system, the part that promotes rest and digestion. When your heart rate slows, that's often the vagus nerve at work. When you take a deep breath and feel yourself relax, the vagus nerve is mediating that response. It carries signals from your gut to your brain, contributing to that gut feeling you get about situations. Increasingly, researchers are discovering that the vagus nerve plays a crucial role in emotional regulation and mental health. Stimulating the vagus nerve, sometimes electrically, sometimes just through deep breathing or meditation, can reduce anxiety, improve mood, and even decrease inflammation throughout the body. This wandering nerve connects your conscious mind to your unconscious body, carrying messages in both directions. These are maintaining the constant conversation between brain and body that keeps you alive and feeling. Your pain signals travel slower than your touch signals, which is why you feel the impact before the pain when you stub your toe. Your nervous system has multiple communication networks, and they operate at different speeds. When you stub your toe, an experience we've probably all had, there's a strange delay between feeling the impact and feeling the pain. You feel the pressure of contact almost instantly, but the pain arrives a split second later. This isn't your imagination. It's a consequence of how different nerve fibers work. Touch signals travel along thick, heavily myelinated nerve fibers that conduct electricity very quickly, those signals racing along at up to 268 miles per hour. Pain signals, particularly the dull, aching kind of pain, travel along thinner, less insulated fibers that conduct much more slowly. Some pain signals crawl along at barely 2 miles per hour. So when you stub your toe, your brain gets a brief warning. Something has made contact with your toe before it gets the full report. And it hurt. It hurt quite a lot, actually. This delay can be disconcerting. That moment of knowing something bad has happened but waiting for the pain to arrive. But it also demonstrates the sophisticated architecture of your sensory system, with different types of information traveling along different pathways at different speeds, all converging in your brain to create your unified experience of sensation. Neuroplasticity means your brain can reorganize itself throughout your entire life, not just in childhood. For most of the 20th century, neuroscientists believed that the brain was essentially fixed after childhood. You were born with your neurons, they made their connections during development, and after that the structure was set. Learn what you could while young, because the window closes. This was accepted wisdom, taught in textbooks, considered settled science. It was completely wrong. Your brain reorganizes itself throughout your entire life. Elderly people learning new languages grow new neural connections in language areas. Musicians who practice an instrument show thickening in regions that control finger movement. London taxi drivers, who must memorize the city's complex streets, develop enlarged hippocampi, the brain region for spatial memory. Even after strokes, the brain can sometimes reroute functions, recruiting new regions to take over for damaged ones. This neuroplasticity means you're never too old to learn, never too set in your ways to change. Your brain remains responsive to experience throughout life. The 80-year-old learning piano is literally growing new neural pathways, forming new synapses, reshaping their brain's physical structure. It happens more readily when you're young, yes, but it never stops entirely. Right now, as you read these words, as you think these thoughts, your brain is changing, adapting, becoming slightly different than it was moments ago. You're always, in the most literal sense, a work in progress. The left and right hemispheres of your brain are connected by about 200 million nerve fibers called the corpus callosum. Your brain is split down the middle into two hemispheres, left and right, each one containing structures that mirror the other side. But these two halves aren't isolated. They're connected by a thick bundle of about 200 million nerve fibers called the corpus callosum, a bridge of neural tissue that allows the two hemispheres to communicate and coordinate their activities. This connection is essential for integrating information. Your left hemisphere might be processing language while your right hemisphere processes spatial relationships, but you experience these as unified aspects of a single thought. The corpus callosum is what allows this integration, letting the two halves of your brain work as one coherent system rather than two separate computers. In rare cases, usually to treat severe epilepsy, surgeons have cut the corpus callosum, creating what's called a split brain patient. The results are fascinating and sometimes unsettling. These individuals can function normally in most situations, but careful testing reveals that their two hemispheres can't share information. Show something to their left eye only, and they can draw it with their left hand but can't name it verbally. It's as if they have two separate streams of consciousness that can't quite communicate. The corpus callosum, that bridge of 200 million fibers, is what makes you feel like one unified person rather than two minds sharing a body. Chronic stress can actually shrink the hippocampus, the brain region responsible for memory and learning. Your experiences don't just affect how your brain functions, they can change its physical structure. Chronic stress, the kind that persists for weeks or months, triggers the release of stress hormones like cortisol. And sustained exposure to cortisol can actually damage neurons in the hippocampus, the seahorse-shaped structure deep in your brain that's crucial for forming new memories and learning new information. Studies have shown that people who experience chronic stress, whether from difficult jobs, traumatic situations, or ongoing life challenges, often have smaller hippocampi than people who don't. This isn't just correlation. Animal studies have demonstrated the mechanism directly. Prolonged stress literally shrinks this brain region, and with it, your capacity for memory and learning can decline. But here's the hopeful part. The brain's plasticity works in both directions. When stress is reduced, the hippocampus can recover. It can regrow, regenerate neurons, restore its volume. People who learn stress management techniques, who address the sources of chronic stress in their lives, can see their hippocampi return to normal size. Your brain is more resilient than you might think. Even damage that seems structural, permanent, can sometimes be reversed. The same plasticity that makes you vulnerable to harm also gives you the capacity to heal. Your nervous system has its own pain sensors in the body, but the brain itself can't feel pain. Here's a strange paradox. Your brain processes all the pain signals from your entire body, creating the conscious experience of pain, managing your responses to injury. And yet the brain itself has no pain receptors. Brain tissue cannot feel pain. You could touch an exposed brain, even cut into it, and the person would feel pressure but not pain. This is why brain surgery can sometimes be performed on awake patients. Surgeons need to map critical regions, language areas, motor control centers, and the best way to do that is to stimulate the brain while the patient is conscious and can report what they experience. the patient feels the pressure of instruments, the strange sensations as different areas are stimulated, but not pain from the tissue itself. The outer coverings of the brain, the meninges, do have pain sensors, which is why headaches hurt. And the blood vessels in the brain are sensitive, but the neural tissue itself, the substance that processes everyone else's pain signals, is paradoxically immune to its own warnings. It's as if the brain, so carefully protected by skull and barrier, was designed on the assumption that if something reaches the brain tissue itself, pain warnings are already too late to matter. A strange oversight, yeah, perhaps, or maybe an elegant simplification. The tissue that interprets pain doesn't need to generate it. Reflexes bypass your brain entirely, using only the spinal cord to process information and respond. When you touch something hot, your hand pulls away before you're consciously aware of the heat. That's not an exaggeration or a trick of perception. It's literally true. The signal doesn't have time to travel all the way to your brain, be processed, and send a response back. Instead, the signal takes a shortcut. Sensory neurons in your hand detect the heat and send signals to your spinal cord. There, in the spinal cord itself, interneurons process this information and immediately send signals back to motor neurons, commanding your muscles to contract and pull your hand away. The entire circuit, sensation, processing, response, happens at the spinal level. Your brain learns about it afterward, receiving the signals slightly later, becoming conscious of what your spinal cord has already handled. This is why reflexes are so fast. They bypass the relatively slow process of conscious decision-making. Your spinal cord takes executive action, makes the call to protect you, and informs your brain about it later. It's a reminder that your consciousness isn't actually in charge of everything. Much of your nervous system operates autonomously, yet making decisions and taking actions without consulting the part of you that thinks it's in control. Right now, your spinal reflexes are monitoring your body, ready to act in milliseconds if needed, a silent guardian that never sleeps. The human nervous system uses the same basic neurotransmitters as insects and worms, chemical languages that evolved over 500 million years ago. The chemical vocabulary your neurons use to communicate is ancient, far more ancient than humanity, and ancient even compared to most of the structures of the modern brain. Neurotransmitters like serotonin, dopamine, and acetylcholine aren't human innovations. They're chemical languages that evolved more than 500 million years ago, before the Cambrian explosion, before most of the animal forms we recognize today even existed. A fruit fly uses serotonin to regulate its mood and behavior. A worm uses dopamine in its simple nervous system. Even jellyfish, which diverged from our lineage more than 600 million years ago, use some of the same neurotransmitters we do. Evolution found chemical signaling systems that worked and has been using them ever since, across vastly different organisms, in vastly different contexts. This means that when you take a medication that affects serotonin levels, you're manipulating a chemical system that predates almost everything we think of as distinctly human. When you feel the rush of dopamine from accomplishing something, you're experiencing a chemical signal that insects also use, translated through your more complex nervous system into human emotion. We are, in a very real sense, using ancient molecular tools, evolutionary hand-me-downs that have been refined and repurposed, but never fundamentally replaced. Your brain creates new neurons in at least two regions throughout your life, the hippocampus and the olfactory bulb. For decades, the dogma was absolute. You're born with all the neurons you'll ever have, and from birth onward, it's all downhill. Neurons dying, none being replaced. This was taught in every neuroscience textbook, accepted as fundamental truth. And then, in the 1990s, researchers discovered it was wrong. Your brain does create new neurons even in adulthood. It happens in at least two regions, the hippocampus, where new memories form, and the olfactory bulb, which processes smell. Thousands of new neurons are born each day in these regions, migrating to their destinations, integrating into existing circuits, becoming functional parts of your nervous system. This neurogenesis seems to be important for certain kinds of learning and for emotional regulation. Exercise increases it. Stress decreases it. Antidepressants may work partly by promoting neurogenesis. You are, quite literally, not the same person neurologically that you were a year ago. Some of the neurons in your hippocampus right now didn't exist last year. They're new cells, fresh additions to ancient circuits, young neurons in an old brain. The discovery overturned fundamental assumptions about the nervous system. It's a reminder that the brain keeps some of its secrets well, and that what we think we know with certainty can turn out to be profoundly incomplete. Sensory neurons can detect a stimulus as small as a single photon of light or a molecule of scent. Your nervous system is sensitive to an almost unbelievable degree. The rod cells in your retina, the sensory neurons responsible for vision in dim light, can respond to a single photon, the smallest possible unit of light. One photon, the quantum minimum. Your eye can, in principle, detect the arrival of a single particle of light. Similarly, olfactory neurons in your nose can detect individual molecules of scent. When you smell something, you're detecting chemical molecules floating through the air, and your olfactory system is sensitive enough to respond to just a few molecules binding to receptors. Some estimates suggest you can detect certain odors at concentrations of just a few parts per trillion. This extreme sensitivity means your nervous system is constantly operating at the edge of what's physically possible to detect. You're perceiving stimuli at nearly the quantum level, and your sensory neurons amplifying signals from single molecules or photons into electrical impulses that travel to your brain. It's almost miraculous. The ability to detect such vanishingly small signals in the midst of all the noise and chaos of the world. Your senses aren't perfect, of course, but they're far more remarkable than we usually appreciate. Right now, in the dim light of evening, your rod cells might be responding to handfuls of photons, creating vision from almost nothing. The peripheral nervous system includes 43 pairs of nerves, 12 from your brain and 31 from your spinal cord. Your brain and spinal cord make up the central nervous system, the command center. But information needs to flow between this center and the rest of your body, and that's the job of the peripheral nervous system. 43 pairs of nerves that branch out to reach every part of you from scalp to toes. 12 pairs emerge directly from your brain, the cranial nerves. They handle your senses and movements in your head and neck, vision, hearing, smell, taste, eye movements, facial expressions, swallowing. The other 31 pairs emerge from your spinal cord at different levels, each one serving a specific region of your body. Nerves from your neck reach your arms and hands. Nerves from your lower back reach your legs and feet. These peripheral nerves form a vast communication network, a biological telecommunication system that maintains constant contact between your central command and your distant territories. Sensory information flows inward: touch, temperature, pain, position. Motor commands flow outward: contract this muscle, relax that one. It's all coordinated through these 43 pairs of nerves, a network that's been with you since before birth, connecting every part of you into one integrated system. You are, in this sense, held together not by belief or will, but by these physical cables of neural tissue, maintaining the communication that makes you feel like one unified being. Your brain's neurons use about 10 watts of power, roughly the same as an energy-efficient LED light bulb. Consciousness runs on about as much power as a reading light. Your neurons, all 86 billion of them firing and processing and maintaining their electrical gradients, consume roughly 10 to 20 watts of power. That's remarkably little, considering what they accomplish. Your brain handles vision, hearing, language, memory, emotion, movement control, and countless unconscious processes, all while using less energy than a dim light bulb. Compare this to artificial neural networks, the computer systems inspired by biological brains. Even simple AI systems can require hundreds of watts, thousands of watts, sometimes megawatts of power to perform tasks your brain handles effortlessly. Yeah, the most powerful supercomputers consume millions of watts to simulate neural networks far simpler than your brain. This efficiency comes from biology. Neurons operate at low voltages, waste minimal energy as heat, and use chemical processes that digital computers can't replicate. Your brain is proof that intelligence doesn't require enormous energy expenditure. It requires the right architecture, the right organization, billions of simple components working together in sophisticated ways. As you drift towards sleep tonight, your brain will reduce its energy consumption even further, entering low-power modes while still maintaining essential functions. 10 watts of power creating the entire universe of your experience. Its efficiency beyond anything human engineering has yet achieved. The neurons in your retina are actually part of your central nervous system. Your eyes are technically outgrowths of your brain. Your eyes aren't really separate from your brain. During development, they form as outgrowths of the developing brain itself, extending forward to create the structures we recognize as eyes. The retina, the light-sensitive tissue at the back of your eye, is actually brain tissue. The neurons there, the ones that detect light and begin processing visual information, are part of your central nervous system, not your peripheral nervous system. This is why the retina is so remarkable. It doesn't just detect light, it processes visual information, extracting edges, and detecting motion, adjusting for brightness, all before the signals even leave your eye. When you look at something, you're not just seeing with your eyes, you're seeing with brain tissue that extends into your eye sockets, processing visual information at the source. This also explains why eye damage can be so difficult to repair. Retinal neurons, like brain neurons, generally don't regenerate. When they're damaged, they're gone. You're losing actual brain tissue, not just peripheral sensors. It's a reminder that what we think of as separate organs, brain, eyes, spinal cord, are really all part of one continuous system. Divisions we impose for convenience, but that biology doesn't entirely respect. When you close your eyes tonight, when you drift towards sleep and vision fades, you're quieting not just your eyes, but portions of your brain itself. Temperature receptors in your skin are actually just pain receptors that trigger at specific temperatures. What you experience as cold or hot isn't really a separate sense from pain, it's a specialized type of danger detection. The receptors in your skin that respond to temperature are actually variants of pain receptors, nociceptors, that have been tuned to activate at specific thermal thresholds. Some trigger when temperatures drop below about 15 degrees Celsius, 59 degrees Fahrenheit, others when they rise above 43 degrees Celsius, 109 degrees Fahrenheit. This makes sense when you think about it. Extreme temperatures are dangerous. They can damage tissue, denature proteins, cause injury. Your nervous system treats temperature as a warning system, alerting you when conditions move outside the safe range. The pleasant warmth of a hot drink or the refreshing cool of a breeze, these are just mild activations of the same sensors that scream in agony when you touch a hot stove or hold ice too long. There's no fundamental distinction between this is hot and this is painfully hot. It's a continuum, different intensities of the same signal. Your brain interprets the strength of the signal and decides how to make you feel about it. Gentle warmth feels pleasant, intense heat feels threatening, but both are flowing through the same neural pathways, different degrees of the same fundamental warning. Pay attention. This temperature matters. Right now, as you lie in bed, your temperature sensors are quietly monitoring the boundary between your skin and the world, making constant small adjustments to keep you comfortable without ever needing to disturb your consciousness. Your brain generates theta waves during deep relaxation and meditation, the same waves present during REM sleep. Your brain has rhythms, electrical oscillations that sweep across neural tissue in coordinated waves. Different mental states produce different patterns. During alert, active thinking, your brain produces fast beta waves. During calm, relaxed awareness, it generates slower alpha waves, and during deep relaxation or meditation, it produces even slower theta waves, rhythmic oscillations of about 4 to 8 cycles per second. What's fascinating is that these same theta waves appear during REM sleep, the stage where vivid dreaming occurs. Experienced meditators can essentially induce a sleep-like brain state while remaining conscious. Their brains show the same electrical patterns as dreaming sleep, but they're awake, aware, observing the patterns of their own thoughts. This suggests something profound about the relationship between meditation and dreaming, between deep relaxation, and the unconscious processing that happens during sleep. Maybe meditation is a way of consciously entering states that usually belong to sleep, of making the unconscious processes temporarily visible. Your brain is capable of these theta rhythms right now. They'll emerge naturally as you drift towards sleep, but with practice. Some people can access them while still awake, existing in a liminal state between waking and dreaming, consciousness and unconsciousness blurred at the edges. The nervous system is the only organ system that uses both electrical and chemical signaling equally. Your body uses many signaling systems. Hormones carry chemical messages through the bloodstream. Mechanical pressure opens and closes valves. But the nervous system is unique in using both electricity and chemistry as equal partners, neither one dominant, both essential to its function. Within a single neuron, information travels electrically. Action potentials racing down axons at high speed, electrical pulses that can cross the length of your body in fractions of a second. But between neurons, information travels chemically, neurotransmitters released into synapses, drifting across nanoscale gaps, binding to receptors on the receiving cell. Electrical for speed and distance, chemical for precision and modulability. This hybrid system gives the nervous system extraordinary versatility. Electrical signals are fast and reliable, good for reflexes and rapid responses. Chemical signals can be amplified, filtered, modulated, even reversed, good for learning and adaptation. The combination allows for circuits that are both fast and flexible, capable of hardwired reflexes and also plastic learning. It's like having both digital and analog systems working together, each compensating for the other's limitations. Where your thoughts right now are flowing through this hybrid network. Electrical spikes triggering chemical releases, triggering more electrical spikes, a cascade of energy and molecules creating the stream of consciousness. Proprioception, your sense of where your body parts are in space, comes from specialized neurons in your muscles and joints. Close your eyes for a moment and touch your nose. You can do it easily, accurately, without seeing your hand or your face. You know where your nose is and where your hand is, even in complete darkness. This is proprioception, your sense of your body's position in space, and it comes from specialized sensory neurons embedded in your muscles, tendons, and joints. These proprioceptors constantly monitor muscle length, tension, and the angles of your joints. They send a continuous stream of information to your brain, creating an internal map of your body's configuration. You don't consciously process this information most of the time, but it's always there, always updating, allowing you to move through the world with confidence. People who lose proprioception through certain neurological conditions find movement almost impossible. Without this internal sense, they must watch their limbs constantly, using vision to substitute for the missing sensory information. It reveals how much we depend on senses we barely acknowledge. Right now, even lying still, your proprioceptors are active, monitoring your position, maintaining your awareness of where you are in space. It's one of those background senses that works so well you never think about it, until you imagine trying to exist without it. Your nervous system can distinguish between your own touch and someone else's touch. You can't tickle yourself. It's not that you're not ticklish. It's that your brain cancels out the sensation when you try to tickle yourself. Your nervous system can distinguish between sensations you cause and sensations caused by external sources, and it treats them differently. This happens through a process called sensory attenuation. When you move, your brain generates a prediction of the sensory consequences, what you should feel as a result of your own actions. When the actual sensation arrives, it's compared to the prediction. If they match, the sensation is dampened, reduced in intensity. This is why you can't surprise yourself with your own touch. But when someone else touches you, there's no prediction. Your brain receives unexpected sensory input and processes it fully without attenuation. This is why a light touch from another person can feel intense, even ticklish, while the same touch from yourself feels muted. Your nervous system is constantly trying to distinguish self from other, expected from unexpected, maintaining a boundary between what you cause and what the world imposes on you. It's a subtle form of self-awareness, written into the sensory processing itself, your nervous system's way of knowing where you end and the rest of the world begins. The enteric nervous system in your gut can produce 95% of your body's serotonin. Here's something that might change how you think about mood and emotion. About 95% of your body's serotonin, that neurotransmitter most strongly associated with happiness and well-being, is produced not in your brain, but in your gut. The enteric nervous system, that network of 500 million neurons lining your digestive tract, is the primary serotonin factory in your body. Most of this gut serotonin doesn't actually cross into your brain. The blood-brain barrier blocks it. It stays in your gut, where it regulates digestion, intestinal movement, and immune function. Your brain makes its own serotonin separately in much smaller quantities. But the fact remains, the neurotransmitter we most associate with brain chemistry is produced primarily in the intestines. This might help explain the complex relationship between gut health and mental health, why digestive disorders often come with mood disturbances, why anxiety affects your stomach, like why antidepressants sometimes cause gastrointestinal side effects. Your gut and your brain are in constant communication, sharing neurotransmitters, influencing each other's function. You are not just a brain piloting a body, you're an integrated system where mood, digestion, emotion, and physical sensation are deeply intertwined. As you settle into rest tonight, your gut's nervous system continues its work, producing the chemicals we associate with contentment, and even as your brain begins its slow descent into sleep. Nerve growth factor, a protein essential for neuron survival, was discovered in the 1950s and earned a Nobel Prize in 1986. For a long time, scientists couldn't figure out how neurons knew to survive, to grow, to extend their axons to the right targets. Neurons seem to develop according to some mysterious internal program that no one could identify. Then, in the 1950s, a researcher named Rita Levi-Montalcini discovered something remarkable. A protein that neurons needed to survive, that guided their growth, that told them where to go and when to stop. She called it nerve growth factor, or NGF. Neurons that received NGF survived and flourished. Neurons deprived of it withered and died. It was one of the first clear demonstrations that chemical signals could directly control neural development and survival. The discovery was so important, so foundational to our understanding of how the nervous system develops, that it earned Levi Montalcini the Nobel Prize in 1986. We now know there's a whole family of these neurotrophic factors, chemical signals that support neural health throughout life. They're active in your brain right now, helping neurons survive, promoting the formation of new connections, supporting the plasticity that allows learning. These chemical supporters work quietly in the background, as maintaining the neural infrastructure that makes thought possible. It's a reminder that neurons aren't self-sufficient, they need support, nourishment, chemical encouragement to thrive. Your olfactory neurons are the only neurons that are regularly replaced throughout your life, renewing about every 30 to 60 days. Almost all your neurons are as old as you are, irreplaceable cells that have been with you since before birth. But there's one exception: your olfactory neurons. The cells in your nose that detect smells are continuously replaced throughout your entire life. They last about 30 to 60 days before dying and being replaced by new neurons born from stem cells. This makes sense when you consider their position. Olfactory neurons are the only neurons directly exposed to the outside world. They stick out into your nasal cavity, directly encountering air, dust, chemicals, pathogens, everything you breathe. This exposure makes them vulnerable to damage, so your body maintains a population of stem cells that can generate fresh olfactory neurons as needed. It's a strange thought. The neurons detecting the smell of your coffee this morning are different cells than the ones that detected the smell of your coffee a few months ago. You're experiencing the world through a constantly refreshed sensory system. New neurons learning the same odors the old ones knew. This renewal might be why smell remains relatively intact as we age. While other senses decline, your olfactory system has built-in regeneration, a biological insurance policy against the inevitable wear and tear of existing at the boundary between your body and the world. The action potential, the electrical spike that travels down a neuron, follows an all-or-nothing principle. Neurons communicate in a binary language. When a neuron fires, it generates an action potential, a spike of electrical activity that races down its axon. And this spike follows what's called an all-or-nothing principle. A neuron either fires completely or doesn't fire at all. There's no such thing as a partial action potential, no dimmer switch, no gradual increase. It's like a gun. You either pull the trigger or you don't. This seems limiting, doesn't it? How can a binary system create the infinite richness of human experience? The answer lies in patterns and rates. A neuron can fire once or many times. It can fire rapidly or slowly. In networks of neurons can create complex rhythms, synchronizations, cascades of activity. From simple on-off signals organized in intricate patterns across billions of neurons emerges everything you think and feel. It's a bit like digital computers, which also use binary signals, ones and zeros, yet can produce symphonies, paintings, simulations of entire worlds. The complexity comes not from the individual signals, but from their organization, their timing, their patterns. Your thoughts right now are patterns of action potentials, billions of binary signals organized into something that feels continuous, analog, seamlessly flowing. Consciousness emerges from discrete spikes, the way a movie emerges from discrete frames, a smoothness created from countless individual moments. Your brain's prefrontal cortex, responsible for decision-making and impulse control, doesn't fully mature until your mid-20s. There's a region right behind your forehead, the prefrontal cortex, that's responsible for some of your most distinctly human capabilities. Planning for the future, controlling impulses, weighing consequences, regulating emotions, making complex decisions. And this region, remarkably, is the last part of your brain to fully mature. It doesn't finish developing until you're in your mid-20s, sometimes later. This means that teenagers and young adults are, in a very real sense, near operating with incomplete neural equipment. Their prefrontal cortex is still forming connections, still myelinating axons, still figuring out how to properly regulate the more impulsive, emotional parts of the brain. It's not a character flaw or a choice, it's neurobiology. Understanding this changes how we might think about adolescence and young adulthood. That period of risk-taking, emotional intensity, questionable decisions, it's not just immaturity. It's a brain that's literally still under construction, particularly in the regions that modulate behavior and foresee consequences. If you're past your mid-20s, your prefrontal cortex has finally finished its long development. If you're younger, it's still growing, still learning, still becoming what it will eventually be. Either way, it's a reminder that we're all works in progress, our very neural architecture evolving across decades. Schwann cells in your peripheral nervous system can guide regenerating nerve fibers back to their original targets. When a peripheral nerve is damaged, cut or crushed, something remarkable can happen. The nerve can regenerate, slowly growing back along the path it once travelled. And guiding this regrowth are specialised cells called Schwann cells, which form the myelin insulation around peripheral nerves. When a nerve is injured, the Schwann cells don't just die off, they survive, lining up along the original path of the nerve like breadcrumbs in a forest. They secrete chemical signals that attract the regenerating nerve fibre, guide it in the right direction, encourage it to keep growing. It's like having microscopic GPS systems built into your nervous system, remembering where each nerve fibre is supposed to go. This guidance isn't perfect. Nerve fibres can get lost, connect to the wrong targets, or fail to regenerate at all, but when it works, it's a small miracle of biological memory. Cells that once insulated a nerve now serve as guides for that nerve's rebirth, maintaining a kind of structural memory of what used to be. As you rest tonight, if you have any healing nerves in your body, Schwann cells are working in the darkness, patiently guiding regenerating fibers millimetre by millimetre back toward their destinations, a slow restoration happening beneath your awareness. The sodium-potassium pump in neurons is constantly working to maintain electrical potential, using about 25% of your body's ATP energy. Your neurons aren't just sitting there waiting to fire. They're constantly working, even at rest, maintaining the electrical gradient that makes firing possible. This work is done by tiny molecular machines called sodium-potassium pumps embedded in every neuron's membrane, constantly pumping sodium ions out and potassium ions in. These are maintaining the electrical charge difference that allows neurons to generate signals. This maintenance is expensive. These pumps consume about 25% of all the ATP energy your cells produce. A quarter of your body's cellular energy budget goes just to keeping neurons ready to fire. Not to actual thinking, not to processing information, just to maintaining readiness. It's like keeping an engine idling, using fuel even when you're not driving anywhere. This is part of why the brain is so metabolically demanding, why it needs so much oxygen and glucose despite its small size. The nervous system is never truly at rest. Even in sleep, even in the quietest moments, your neurons are burning energy to maintain their electrical potential, staying poised on the edge of firing, ready to spring into action at a moment's notice. It's a kind of constant vigilance, a biological commitment to readiness that never stops. We consuming energy just to preserve the possibility of thought. Astrocytes, a type of glial cell, can listen to neural conversations and actually modulate how neurons communicate. For decades, we thought neurons did all the important work and glial cells just supported them. But astrocytes, star-shaped glial cells that outnumber neurons in many brain regions, turn out to be active participants in neural communication. They have extensions that wrap around synapses, the junctions between neurons, and they can listen to the chemical conversations happening there. More than just listening, astrocytes can respond. They can take up excess neurotransmitters, cleaning the synapse. They can release chemicals that strengthen or weaken synaptic connections. They can even modulate the flow of blood to active brain regions, ensuring neurons get the resources they need. In essence, astrocytes are regulating neural conversations, deciding which ones should be enhanced and which should be dampened. This means that what we call neural computation, the processing that creates thoughts and memories, isn't just happening in neurons. It's happening in a partnership between neurons and astrocytes, a collaboration where glial cells actively shape neural function. Your thoughts aren't purely neuronal. They're emerging from interactions between multiple cell types, working together in ways we're only beginning to understand. The brain is even more complex than the number of neurons suggests. It's not just 86 billion neurons, but 86 billion neurons in constant dialogue with even more glial cells, all shaping each other's behavior. Your nervous system processes visual information so fast that you can recognize a face in about 170 milliseconds. Look at someone's face, and you know, almost instantly, whether you recognize them. That recognition happens in about 170 milliseconds, less than a fifth of a second. Faster than you can blink, and yet, in that brief time, your visual system has processed an enormous amount of information. Light hits your retina, triggering millions of photoreceptors. Signals flow through multiple layers of retinal neurons, extracting edges, contrasts, movement. Information races along the optic nerve to your brain, to the visual cortex at the back of your head. From there, signals fan out to specialized regions. Some detect faces, others detect emotional expressions. Others access memory to determine if this face is familiar. All of this happens in 170 milliseconds. Your visual system is processing millions of data points, the curve of a cheek, the spacing of eyes, the shape of a smile, comparing them to stored memories, integrating emotional context, preparing appropriate social responses. It's computational work that would challenge even sophisticated computers, and your brain does it effortlessly, continuously, faster than conscious thought. Right now, if you're reading this by the light of a screen or lamp, your visual system is performing these miracles of processing every moment, creating the seamless experience of vision that feels so simple, but is actually extraordinarily complex. The neurotransmitter glutamate is used in about 90% of excitatory synapses in your brain. Your brain's primary go signal, the neurotransmitter that tells neurons to fire, to activate, to pass messages along, is a simple molecule called glutamate. About 90% of the excitatory synapses in your brain use glutamate as their primary chemical messenger. It's the workhorse neurotransmitter, the common language of activation. Glutamate is essential for learning and memory. When you form a memory, glutamate strengthens the synapses involved, making those neural connections more likely to activate in the future. Long-term potentiation, the basic mechanism of learning, depends on glutamate signaling. Every skill you've learned, every fact you've memorized, every association you've formed, all of it mediated by glutamate binding to receptors, strengthening connections. But glutamate is also dangerous in excess. Too much glutamate can overexcite neurons, causing them to fire uncontrollably until they exhaust themselves and die. A process called excitotoxicity. This happens in strokes, when damaged tissue releases massive amounts of glutamate, killing surrounding neurons. Your brain carefully regulates glutamate levels, balancing the need for excitation against the danger of overexcitation. It's a reminder that neurotransmitters aren't inherently good or bad. They're tools, and like any tool, their effect depends on dose, context, and control. GABA, the brain's main inhibitory neurotransmitter, is equally important. It's the stop signal that prevents neural chaos. If glutamate is the brain's gas pedal, GABA is the brake. GABA, gamma-aminobutyric acid, is the primary inhibitory neurotransmitter in your brain. The chemical signal that tells neurons not to fire, to quiet down, to reduce their activity, and it's just as essential as excitation. Without inhibition, your brain would spiral into chaos. GABA prevents runaway excitation. It creates the pauses between thoughts, the silence between words, the rest between movements. Many anti-anxiety medications work by enhancing GABA signaling, increasing inhibition throughout the brain, creating a general calming effect. Alcohol does something similar, which is why drinking makes people relaxed. It's enhancing inhibition, dampening neural activity. But GABA does more than just prevent chaos. It's essential for precise neural processing. Like silence in music, inhibition gives structure to neural activity. It sharpens signals, prevents irrelevant neurons from firing, creates the precise patterns that carry information. Your brain is constantly balancing excitation and inhibition, glutamate and GABA, go signals and stop signals. Right now, as you settle towards sleep, GABA is likely increasing its influence, inhibiting the circuits that keep you alert, allowing the slow rhythms of sleep to emerge. Without this inhibitory messenger, rest would be impossible. Your brain consolidates memories during sleep by replaying neural firing patterns from the day at high speed. While you sleep, your brain is far from idle. It's reviewing the day's experiences, literally replaying them. Neural firing patterns that occurred during waking hours, when you learned something new, experienced something memorable, fire again during sleep, but faster, compressed in time. It's like watching a video in fast forward, the essential patterns preserved but accelerated. This replay seems to be how memories transition from temporary storage in the hippocampus to more permanent storage in the cortex. During sleep, your brain is deciding what's worth keeping, then strengthening the synapses associated with important experiences, allowing less important ones to fade. It's editing the day's footage, deciding what makes it into long-term memory and what gets deleted. This is why sleep is so important for learning. You can study all you want while awake, but the real consolidation happens during sleep. Students who sleep after learning remember more than students who stay awake. Your brain needs these quiet hours to process, to integrate, is to move experiences from short-term to long-term storage. Tonight, as you drift off, your brain will begin this work, reviewing your day, strengthening memories, integrating new information with old, quietly organizing the vast library of your experience while you dream. The dorsal root ganglion contains the cell bodies of sensory neurons, essentially serving as sensory headquarters outside the spinal cord. Your nervous system has an unusual architectural feature. Most neurons keep their cell bodies, the part that contains the nucleus and does the metabolic work, inside the central nervous system, in the brain or spinal cord. But sensory neurons are different. Their cell bodies cluster in structures called dorsal root ganglia, small nodules that sit just outside the spinal cord, one pair for each spinal segment. It's a strange design choice. Why keep sensory neuron cell bodies outside the protective confines of the spinal cord? No one knows for certain, but it might have to do with development, the way the nervous system forms during embryonic growth, or it might serve some functional purpose we don't yet understand. What it means, practically, is that you have clusters of neural cell bodies sitting outside your central nervous system, vulnerable to injury but also accessible in ways that central neurons aren't. These ganglia are sensory headquarters, coordinating the flow of information from your skin, muscles and organs to your spinal cord. Nay, they're working right now, processing touch and pain and proprioception, sending carefully organized signals inward to the spinal cord, maintaining the constant flow of sensory information that tells you what your body is experiencing. Myelination, the process of insulating nerve fibers, continues into your 30s in some brain regions. Your brain is still physically developing well into adulthood. One of the last processes to complete is myelination, the wrapping of nerve fibers in fatty insulation that speeds up signal transmission. While much myelination happens in childhood, some brain regions continue myelinating into your 20s and 30s, particularly in the prefrontal cortex and other areas involved in complex reasoning and self-control. This means your brain is literally still under construction into your 4th decade of life. neural signals are getting faster, more efficient, or better coordinated. The white matter, those myelinated tracts connecting different brain regions, is still increasing in volume, improving the speed and efficiency of long-range communication in your brain. This extended development might explain why people often feel they continue maturing psychologically and emotionally through their 20s and into their 30s. It's not just experience and learning, it's actual physical changes in brain structure, neural circuits becoming faster and more efficient. If you're in your 30s or beyond, your myelination is likely complete. If you're younger, your brain is still fine-tuning its wiring, still optimizing the speed of communication between distant regions. Either way, it's a reminder that development is a long process, that becoming fully yourself takes decades of both experience and biological maturation. Optogenetics allows scientists to control specific neurons with light, essentially letting them switch thoughts on and off. In the past 2 decades, neuroscience developed a technique that sounds like science fiction: optogenetics. Scientists can genetically modify neurons to respond to light, then use precisely targeted light beams to activate or silence specific groups of neurons. It's like installing light switches in the brain, allowing researchers to turn specific neural circuits on or off with remarkable precision. Using optogenetics, researchers have made mice remember things that never happened, created false memories by activating specific hippocampal neurons. They've made animals suddenly feel fear or pleasure by activating emotional circuits. They've restored movement in paralyzed animals by activating motor neurons with light. It's an unprecedented level of control over the nervous system. The technique reveals something profound. Mental states aren't mystical or irreducible. They're patterns of neural activity, and if you can control the pattern, you can control the state. You activate the right neurons in the right sequence, and you can create memory, emotion, perception, apparently from nothing. It's both exciting and unsettling. Proof that consciousness and experience are in some sense just physics and chemistry, patterns that can be manipulated if you have the right tools. Your thoughts right now feel private, internal, yours. But in principle, with the right technology, they could be read, modified, even created from scratch. Your nervous system can process pain signals from your body, but also generate pain entirely on its own when damaged. Pain is usually a message. Tissue is damaged. Something is wrong. You need to protect this area. But sometimes pain becomes the problem itself. When the nervous system is damaged through injury, disease, or malfunction, it can generate pain signals with no underlying injury to warn you about. This is chronic pain, and it's the nervous system misfiring, near creating suffering without purpose. Damaged nerves can become hyperactive, firing spontaneously, sending pain signals to the brain even though nothing is actually wrong with the body part that hurts. Or the pain processing centers in the brain and spinal cord can become sensitized, amplifying normal signals into pain, lowering their threshold for activation. What should be a gentle touch becomes agony. What should be painless becomes unbearable. This is why chronic pain is so difficult to treat. You're not fighting an injury. You're fighting the nervous system itself, trying to convince it to stop generating signals that serve no protective purpose. It's a reminder that the nervous system, for all its sophistication, can malfunction in ways that cause genuine suffering. Pain that once helped us survive can, when the system breaks, become a torment with no solution. For those living with chronic pain, the nervous system has become an adversary, a source of constant false alarms that can't be silenced. The axon of a single motor neuron can be over 3 feet long, stretching from your spinal cord to your foot. Consider the logistics of your body. A single neuron in your spinal cord needs to control a muscle in your foot. The cell body sits in your lower spine, but the muscle is 3 feet away, maybe more. So the neuron extends an axon, a single, continuous cellular process that stretches the entire distance. 3 feet of living cellular material, maintained by a single cell carrying signals from your spinal cord to your toes. This makes motor neurons some of the longest cells in your body. They're not just cells, they're cables, biological wires that can stretch for impressive distances. And when they fire, electrical signals race down this entire length in milliseconds, travelling at hundreds of miles per hour, arriving at the muscle almost instantaneously despite the distance. Maintaining such a long structure is challenging. The cell body must manufacture proteins and other materials, then transport them all the way to the distant end of the axon. Damaged regions far from the cell body are difficult to repair. These neurons are architectural marvels, single cells stretched to extreme lengths to bridge the gap between your central nervous system and your far-flung muscles. Right now, your neurons extending from your spine to your feet are maintaining this incredible length, ready to fire the moment you decide to move, sending signals racing down their full extent in a fraction of a second. Dendrites, the branching receivers of neurons, can change shape within hours based on neural activity. Neurons aren't static structures. They're dendrites, the branched extensions that receive signals from other neurons, can grow, shrink, sprout new branches, or retract old ones, all in response to activity. And this happens quickly, sometimes within hours. Learn something new, and dendrites begin sprouting new spines, creating new sites where synapses can form. Stop using a skill, and dendrites retract, eliminating unused connections. This structural plasticity is part of how learning works at the cellular level. Your brain isn't just changing the strength of existing connections, it's physically remodeling the connections themselves. Growing new branches where activity is high, pruning branches where activity is low. It's gardening on a cellular scale. Cultivation and pruning happening constantly based on experience. Time-lapse imaging of neurons shows this process in action. Dendrites extending, exploring, retracting, like plants growing in fast forward. Your brain's physical structure is in constant flux. Dendrites reaching out and pulling back. The shape of individual neurons responding to the patterns of signals they receive. Right now, as you process these ideas, some of your dendrites might be sprouting new spines, creating the physical basis for tomorrow's memories. Your brain is sculpting itself in real time, experience directly shaping structure. The sympathetic nervous system prepares you for action, while the parasympathetic system calms you down. Your autonomic nervous system has two branches, so working in opposition like a gas pedal and brake. The sympathetic nervous system activates when you need to respond to challenges. It's the fight or flight system. It increases your heart rate, dilates your pupils, redirects blood from digestion to muscles, releases stress hormones. It prepares your body for action, for emergency, for dealing with threats. The parasympathetic nervous system does the opposite. It's the rest and digest system. It slows your heart, promotes digestion, encourages relaxation and recovery. It's active when you're safe, when you can afford to rest, when your body can focus on maintenance rather than emergency response. You're always balanced between these two systems, in a constant negotiation between alertness and relaxation. Right now, as you read this in the evening, preparing for sleep, your parasympathetic system is likely gaining dominance, slowing things down, encouraging rest, telling your body it's safe to let go of vigilance. This balance shifts throughout the day, responding to stress, safety, time of day, countless subtle cues. Your autonomic nervous system is conducting an invisible orchestra, adjusting thousands of parameters to keep you balanced between readiness and rest, between vigilance and recovery. Neurotransmitter receptors on neurons can change in number and sensitivity, a process that takes about a week. Your neurons don't just passively receive neurotransmitter signals, they adjust their sensitivity based on how much signaling they're getting. If a neuron receives too much of a particular neurotransmitter, it can reduce the number of receptors on its surface, making itself less sensitive. If it receives too little, it can add more receptors, making itself more sensitive. This process takes about a week to fully manifest. This is why many psychiatric medications take weeks to show their full effect. Antidepressants, for example, might start blocking serotonin reuptake immediately, increasing serotonin levels in the synapse within hours. But the real therapeutic effect doesn't appear until the receiving neurons have had time to adjust their receptor populations, rebalancing their sensitivity to match the new chemical environment. It's a reminder that the nervous system isn't a fixed circuit. It's adaptive. They are constantly adjusting its own sensitivity based on experience. Too much stimulation leads to desensitization. Too little leads to sensitization. Your neurons are always trying to maintain optimal responsiveness, tuning themselves like instruments to stay properly calibrated. This adaptation happens throughout your nervous system, responding to your experiences, your environment, your habits, slowly adjusting the sensitivity of neural circuits to maintain function, despite constantly changing conditions. Your brain's default mode network activates when you're not focused on anything specific, when you're daydreaming or mind-wandering. When you let your mind wander, when you daydream or just space out, it feels like your brain is resting. But brain imaging reveals something different. A specific network of brain regions becomes active during these unfocused moments. It's called the default mode network, and it's working hardest when you're not working on any particular task. What's this network doing? It seems to be involved in self-reflection, thinking about the future, recalling the past, imagining alternative scenarios, processing social information. It's the network that creates your internal mental life, the stream of thoughts, memories and imaginings that fills your mind when external demands don't occupy it. Interestingly, the default mode network tends to quiet down when you focus on external tasks, then reactivates during rest. It's as if your brain has two modes, one directed outward, engaging with the immediate environment, and one directed inward, exploring your internal mental landscape. Right now, reading these words, your task-focused networks are active. But when you finish reading, when you turn off the light and lie in darkness, your default mode network will likely activate, regenerating the wandering thoughts that accompany the drift towards sleep. What feels like mental emptiness is actually a specific pattern of coordinated neural activity. Oligodendrocytes in the central nervous system can wrap myelin around up to 50 different axons simultaneously. In your brain and spinal cord, the cells that create myelin insulation, oligodendrocytes, are remarkably efficient. A single oligodendrocyte can extend multiple processes, each one wrapping around a different axon, thus creating myelin sheaths for up to 50 different nerve fibers simultaneously. It's like one worker managing 50 different insulation projects at once. This efficiency is necessary. Your central nervous system has billions of axons that need insulation, and oligodendrocytes must provide it. Each oligodendrocyte spreads itself thin, literally, extending tendrils to multiple nearby axons, wrapping each one in the fatty myelin that speeds up signal transmission. Without this efficiency, you'd need impossibly many cells to myelinate your entire nervous system. But this efficiency comes with a vulnerability. If an oligodendrocyte dies or becomes damaged, as happens in diseases like multiple sclerosis, up to 50 different axons lose their insulation simultaneously. Signal transmission slows, neural communication breaks down, and the effects can be devastating. Your nervous system's efficiency, its elegant design of having single cells serve many axons, creates points of vulnerability where damage to one cell affects many circuits. Right now, throughout your brain, oligodendrocytes are maintaining the myelin on thousands of axons each, working quietly to keep your neural signals fast and coordinated. The nervous system develops from a sheet of cells that rolls up into a tube during the 3rd week of embryonic development. In the 3rd week after conception, something remarkable happens. A flat sheet of cells on the surface of the embryo, the neural plate, begins to fold. The edges lift up, curl toward each other and fuse, creating a hollow tube. This neural tube will become your entire central nervous system, your brain and spinal cord. Everything you think and feel will emerge from this structure. The process is called neurulation, and it's one of the most critical events in development. If it doesn't happen correctly, if the tube doesn't close properly, the results can be severe. Spina bifida, where the spinal cord doesn't fully form, or an encephaly, where much of the brain fails to develop. The entire architecture of your nervous system depends on this one origami-like folding event, happening correctly in the 3rd week of development. It's beautiful in its simplicity, a flat sheet becoming a tube, and from that tube, through cell division and migration and differentiation, emerging the entire complexity of the nervous system. Your brain began as a simple geometric transformation, cells rearranging from 2 dimensions into 3, creating the basic structure from which all neural complexity would eventually emerge. Before there was thought, before there was sensation, there was just this. A sheet of cells rolling up, closing, forming a tube in the darkness of the womb. Neurotransmitters are recycled. After they deliver their message, they're often reabsorbed and reused. Chemical signaling could be wasteful. Release neurotransmitters, let them bind to receptors, then what? Throw them away? Make new ones each time? The nervous system has a more elegant solution: recycling. After neurotransmitters are released into the synapse and deliver their message, many of them are taken back up into the releasing neuron through reuptake transporters, packaged back into vesicles, and reused. This recycling is so important that interfering with it has profound effects. Now, many antidepressants work by blocking serotonin or norepinephrine reuptake, forcing these neurotransmitters to linger in the synapse longer. Cocaine blocks dopamine reuptake, creating the intense pleasure and energy that makes the drug addictive. By preventing recycling, these drugs amplify the signals these neurotransmitters carry. It's a reminder that the nervous system is conservative with its resources. Neurotransmitters are expensive to synthesize. We and the nervous system generates trillions of signals every day. Recycling makes this sustainable, allowing the same molecules to be used again and again, carrying message after message. Right now, neurotransmitters that delivered signals moments ago are being packaged back into vesicles, ready to be released again. An endless cycle of use and reuse that makes the constant chemical conversation of the nervous system possible. The reticular formation in your brainstem controls consciousness itself. Damage here can lead to permanent coma. Deep in your brainstem, there's a network of neurons called the reticular formation, a diffuse, complex structure that looks almost unorganized compared to the neat layering of the cortex. But this messy-looking network has one of the most important jobs in the entire nervous system. It controls consciousness. It determines whether you're awake or asleep, alert or drowsy, conscious or unconscious. The reticular formation sends activating signals throughout the brain, maintaining wakefulness. When it's active, you're conscious. When it quiets, you drift towards sleep. If it's damaged through stroke, trauma, or disease, the result can be permanent loss of consciousness, a coma from which no amount of external stimulation can wake you. Your cortex might be intact, all the machinery for thinking and feeling still present, but without the reticular formation to activate it, consciousness doesn't emerge. It's both humbling and frightening. Consciousness, that most essential aspect of your existence, depends on this small, diffuse network of neurons in your brainstem. All your thoughts, memories, personality, none of it matters if the reticular formation isn't working. Right now, as you read this, your reticular formation is maintaining your wakefulness, keeping you conscious. Soon, as you prepare for sleep, it will gradually reduce its activity, allowing consciousness to fade, the world to recede, and sleep to claim you. Synesthesia, where senses blend together, reveals how flexibly the nervous system can wire itself. For most people, Senses are distinct. You hear sounds, see colours, taste flavours, each in its own separate channel. But some people experience synesthesia, where sensory signals cross in unexpected ways. They might see specific colours when they hear certain musical notes. They might taste shapes. They might experience numbers as having inherent colours. 3 is always yellow, 7 is always green. This isn't metaphor or imagination. Brain imaging shows that when synesthetes experience these crossed sensations, multiple sensory regions activate simultaneously. A sound genuinely triggers their visual cortex. A number genuinely activates color processing areas. The neural wiring is literally different, with connections that most people don't have, and creating experiences most people can't imagine. Synesthesia reveals how flexible neural development can be. The same basic brain, the same basic building blocks, can wire itself in radically different ways, creating fundamentally different experiences of reality. What you experience as separate and distinct, others experience as unified and blended, where it's a reminder that your experience of the world, the seemingly objective reality of separate senses, is actually constructed by your particular pattern of neural connections. Change the wiring, and the entire subjective experience changes. Your reality is not the only possible reality, even with the same inputs. Your brain's hippocampus creates cognitive maps of spaces you navigate, with specific neurons firing for specific locations. When you navigate through space, walking through your home, driving to work, exploring a new city, your hippocampus is creating a map. And this isn't a metaphorical map. It's a literal neural representation where specific neurons fire only when you're in specific locations. These place cells activate when you're in your kitchen but not your bedroom, when you're at this intersection but not that one. Researchers discovered this by recording from individual neurons in rats as they explored mazes. Yes, certain neurons would fire only when the rat was in a particular corner, silent everywhere else. Each place had its own cells, creating a neural map of space. Humans have the same system. Your hippocampus contains a representation of every space you know, encoded in the firing patterns of place cells. This is why the hippocampus is essential for spatial memory. Damage it, and people lose the ability to navigate, to remember where things are, to form mental maps of new spaces. London taxi drivers, who must memorize thousands of streets, have enlarged hippocampi, their brains physically growing to accommodate the vast spatial information they need. Right now, your hippocampus contains maps of every place you've been, neural representations of rooms and streets and buildings, ready to activate when you imagine those places or return to them in reality. Your memories of space are written in the language of place cells. Long-term potentiation is the mechanism by which memories are encoded, strengthening synapses that fire together. There's a saying in neuroscience, neurons that fire together, wire together. This captures the essence of long-term potentiation, or LTP, the process by which synapses strengthen when presynaptic and postsynaptic neurons are active simultaneously. It's the cellular basis of learning and memory, the mechanism by which experience physically changes your brain. When two connected neurons fire at the same time repeatedly, the synapse between them strengthens. more neurotransmitter receptors appear on the receiving neuron. The connection becomes more efficient, more likely to trigger firing in the future. This strengthening can last for hours, days, even a lifetime. It's how associations form, how skills develop, how memories solidify. Every memory you have exists as a pattern of strengthened synapses. When you recall a memory, you're reactivating that pattern, causing those specific neurons to fire together again. And each time you recall it, you potentially strengthen the synapses further, or modify them, which is why memories can change over time. Your entire personal history, everything you know and remember, exists as physical modifications to synaptic strength throughout your brain. LTP is the mechanism that makes you you, the cellular process that encodes experience into neural structure. Right now, as you read and think about these ideas, LTP is occurring, synapses strengthening. Tomorrow's memory is being physically written into your brain. Your nervous system maintains a precise body temperature map, knowing exactly how warm or cold each part of you is. Temperature isn't a single sensation, it's a map. Your nervous system knows the temperature of your forehead, your fingertips, your toes, your back, all independently. Temperature receptors throughout your skin send constant updates to your brain, creating a detailed thermal map of your body's surface. This map is precise enough that you can detect which specific part of you is warm or cold without looking. This mapping creates interesting phenomena. Put your left hand in ice water and your right hand in warm water, then put both in lukewarm water. The lukewarm water will feel warm to your left hand and cool to your right hand, and even though it's the same temperature, your nervous system judges temperature relative to recent experience separately for each body part. Your brain uses this temperature map for thermoregulation, deciding when to shiver, when to sweat, when to redirect blood flow. It's monitoring your thermal environment constantly, comparing it to your body's core temperature, making adjustments to keep you in the safe range. Right now, as you lie in bed, your nervous system is tracking the temperature of every part of your skin, noting the warmth where you contact the blanket, the coolness where you're exposed to air, maintaining a complete thermal awareness of your body's boundary with the world. The amygdala processes fear so quickly that you can react to danger before you're consciously aware of it. There are two pathways for fear processing in your brain. The slow pathway sends sensory information through the cortex, where it's carefully analysed and interpreted before reaching the amygdala, your brain's fear centre. This pathway is thorough but takes time. The fast pathway goes directly from sensory organs to the amygdala, bypassing conscious processing entirely. This fast pathway is why you can jump away from a snake before you consciously recognize it's a snake. The amygdala sees something snake-shaped, triggers a fear response, and your body reacts, all before your cortex finishes analyzing the visual input. You jump first, then a split second later become consciously aware of what you're jumping from. It's a survival mechanism. When danger appears, waiting for conscious analysis might be fatal. Better to react immediately to potential threats, even if some turn out to be false alarms. Your amygdala is always scanning for danger, processing sensory input through this express pathway. ready to trigger fear responses before you consciously know why. It's working right now, quietly evaluating your environment, deciding whether you're safe or threatened. In the safety of your bed, it's likely quiet, allowing you to relax, but it never stops watching, never stops being ready to flood you with fear if needed. Neuromelanin, a dark pigment in certain neurons, increases with age and may protect against neurodegenerative diseases. Deep in your brain, in regions like the substantia nigra and locus coeruleus, certain neurons accumulate a dark pigment called neuromelanin. It's similar to melanin in your skin, but it's found only in neurons, and it increases gradually throughout your life. By old age, these neurons can appear almost black under microscopy. We're not entirely sure what neuromelanin does, but evidence suggests it might be protective. It can bind to metals and toxins, potentially sequestering harmful substances away from sensitive cellular machinery. Neurons with high neuromelanin content seem more resistant to certain types of damage. In Parkinson's disease, which involves the death of neurons in the substantia nigra, the neurons that die are specifically the ones with the most neuromelanin. Your brain is literally getting darker as you age, accumulating this mysterious protective pigment in specific neural populations. It's one of many ways your nervous system changes over time, adapting to decades of existence, accumulating both wear and protective mechanisms. The neurons in your substantia nigra right now contain neuromelanin that wasn't there when you were younger, a slow darkening that's been happening throughout your life, quietly, invisibly, possibly protecting you from damage you'll never know you avoided. The blood-brain barrier is so selective that glucose, your brain's main fuel, needs special transporter proteins to cross it. Your brain runs on glucose. It's the primary fuel for neural activity, essential for maintaining consciousness, processing information, keeping neurons alive. But glucose molecules can't simply diffuse from your bloodstream into your brain tissue. The blood-brain barrier blocks them. Instead, special transporter proteins in the blood vessel walls grab glucose molecules and shuttle them across. This seems inefficient, doesn't it? Your brain desperately needs glucose. Glucose is abundant in your blood, yet it requires special transport machinery to access it. But this selectivity is the price of protection. The blood-brain barrier is so strict because the brain is so vulnerable. Better to tightly control everything that enters, even essential nutrients, than to risk letting in toxins or pathogens. Your brain is constantly importing glucose through these transporters, maintaining the energy supply needed for thought and consciousness. When blood sugar drops too low, these transporters can't keep up, and brain function suffers. Confusion, dizziness, difficulty concentrating. Your thoughts, in a very literal sense, run on sugar, carefully imported across a highly selective barrier. Right now, glucose transporters in your brain's blood vessels are working continuously, pulling fuel from your bloodstream by maintaining the energy supply that keeps your neurons firing and your consciousness intact. Your nervous system can detect the difference between a 1-degree temperature change on your skin. The precision of your thermal sensing is remarkable. Your skin can detect temperature changes as small as 1 degree Celsius, sometimes even less in particularly sensitive areas. This sensitivity helps you navigate environments, avoid harm, maintain comfort. A slight change in air temperature, a subtle shift in the warmth of an object you're touching, your nervous system notices. This precision comes from specialized thermoreceptors that respond to specific temperature ranges. Some activate as temperature rises, others as it falls. They're distributed throughout your skin at varying densities. Your face and hands have more than your back or legs. Together, they create a sensitive thermal detection system that constantly monitors your environment. This sensitivity serves survival. Small temperature changes can signal danger, a gradual warming that might lead to burns, a slow cooling that might lead to hypothermia. Your nervous system catches these changes early, when you can still respond before damage occurs. Right now, your thermoreceptors are monitoring the temperature of your environment, noting the warmth of your blanket, the coolness of the air, maintaining your awareness of thermal comfort. Most of this monitoring happens below consciousness, but the information is there, constantly updating, keeping you informed about your thermal world. Neurogenesis in adults was discovered only in the 1990s, overturning decades of scientific dogma. For most of the 20th century, neuroscience's central dogma was clear and absolute. You're born with all the neurons you'll ever have. The adult brain cannot generate new neurons. This was taught in every textbook, repeated by every expert, accepted as fundamental truth. Researchers who suggested otherwise were dismissed or ignored. Then, in the 1990s, evidence became undeniable. Adult brains do generate new neurons, particularly in the hippocampus. The dogma was wrong. For decades, science had been confidently teaching something fundamentally incorrect about the brain. The discovery forced a massive revision of neuroscience, opening new possibilities for understanding learning, memory, and recovery from brain injury. It's a humbling reminder of how much we still don't know, how confident assertions can turn out to be completely wrong. The researchers who overturned this dogma faced skepticism and resistance, yet they were right. Your brain can do something that scientists insisted for decades it couldn't do. It makes you wonder what other settled facts about the nervous system might someday be revealed as misconceptions. Science advances through such overturnings, we old certainties giving way to new discoveries, our understanding deepening through the willingness to question even fundamental assumptions. The claustrum, a thin sheet of neurons beneath the cortex, may act as a conductor coordinating consciousness. Beneath your cortex, there's a thin, mysterious structure called the claustrum, a sheet of neurons that connects to nearly every region of your brain. For a long time, no one knew what it did. It seemed too small and obscure to be important. But some neuroscientists now believe it might play a crucial role in consciousness itself, acting like a conductor coordinating the brain's various regions into unified experience. The claustrum has an unusual connectivity pattern. It sends and receives connections from most of the cortex, creating a hub that could integrate information from many different brain regions. When the claustrum is electrically stimulated in certain ways, people report losing consciousness temporarily. or when it's damaged, strange alterations in consciousness can occur. The idea is that consciousness requires integration. Visual information, auditory information, memories, emotions, all combined into a single unified experience. The claustrum might be where this integration happens, where separate streams of information are coordinated into the seamless sense of experiencing one coherent world. It's speculative, not proven, but intriguing. Your sense of being one unified person, or experiencing one integrated reality, might emerge from this thin sheet of neurons conducting the orchestra of your brain, ensuring all the separate parts play together in harmony. Your brain's neurons consume glucose and oxygen, but produce only water, carbon dioxide, and heat as waste. Thinking is, chemically speaking, a remarkably clean process. Your neurons consume glucose and oxygen, fuel and oxidizer, and through cellular respiration, they produce ATP energy to power their electrical signaling. The waste products are simple: water, carbon dioxide, and heat. No toxic byproducts, no heavy metals, no complex molecules that need elaborate disposal systems. The carbon dioxide dissolves in your blood and gets exhaled through your lungs. The water becomes part of your body's fluid. The heat radiates away or is carried off by blood flow. It's an elegant system. Neurons burn fuel intensely, generating the energy needed for constant electrical activity, yet produce waste that's easy for the body to handle. This efficiency is part of why the brain can operate continuously for decades without accumulating significant metabolic debris. During sleep, glial cells do clear away some metabolic waste products that accumulate during waking hours, but the primary waste, water and carbon dioxide, handles itself. Right now, your neurons are burning through glucose, generating electrical signals, producing these simple waste products that your body quietly eliminates. Consciousness generates heat and exhalation, thought made physical in the warmth of your body and the rhythm of your breath. Microglia, the immune cells of the nervous system, actively prune unnecessary synapses during development and learning. Your brain doesn't just add connections, it also strategically removes them. Microglia, the immune cells of the nervous system, do more than just fight infections. They actively prune synapses, eliminating connections that are weak, unused, or unnecessary. This pruning is essential for healthy brain development and function. During adolescence, massive synaptic pruning occurs. Your brain eliminates roughly half of the synapses it had in childhood, keeping the ones that are strong and frequently used, removing the ones that aren't. Microglia are the gardeners doing this work, sculpting neural circuits by selective elimination. Even in adulthood, they continue pruning, removing synapses during learning, helping to sharpen neural circuits. This means learning isn't just about creating connections, it's also about destroying them. Becoming skilled at something involves eliminating the neural noise, the unnecessary connections, leaving only the efficient pathways. Your expertise in any domain exists partly because microglia have pruned away the synapses you didn't need. Right now, throughout your brain, microglia are surveying synapses and deciding which ones to keep and which ones to eliminate, quietly sculpting your neural circuits while you sleep and wake, shaping your brain through strategic subtraction. The nervous system can create entirely false memories that feel completely real. Your memories aren't perfect recordings of the past. They're reconstructions, assembled each time you recall them from scattered pieces stored across your brain. And during this reconstruction, errors can occur. Details can be added, modified, or changed entirely. You can remember events that never happened, conversations that never occurred, experiences you never had, and these false memories can feel just as vivid and real as true ones. Research has shown this repeatedly. People can be convinced they got lost in a shopping mall as a child, or met Bugs Bunny at Disneyland, or witnessed events they only heard described. The false memory feels genuine, complete with sensory details and emotional resonance. The brain imaging can't distinguish true memories from false ones. They activate the same regions, produce the same patterns. This is deeply unsettling. Your memories, your sense of your own history, the narrative of your life, all of it is potentially unreliable. Every time you remember something, you're actually remembering the last time you remembered it, not the original event. Each recall is an opportunity for modification, for subtle changes that accumulate over time. The past as you remember it might not be the past as it actually was. Your nervous system, trying to piece together fragmented information, sometimes fills in gaps with plausible fictions, creating false memories that integrate seamlessly with true ones. Your brain has dedicated neurons that respond only to specific concepts, like a Jennifer Aniston neuron or a grandmother cell. Deep in your brain, in the medial temporal lobe, these researchers have found neurons that respond to surprisingly specific concepts. Single neurons that fire only when you see pictures of Jennifer Aniston. Other neurons that respond to Halle Berry, or the Sydney Opera House, or your grandmother. These neurons respond to the concept itself, not just the specific image. They'll fire for photographs, drawings, even just seeing the person's name. This seems almost absurd. How can a single neuron represent an entire complex concept? The answer is that it doesn't work alone. These concept cells are part of larger networks, representing ideas through patterns of activity across many neurons. But the fact that single neurons can be so selective, so specific in what they respond to, reveals something profound about how the brain organizes information. Your brain doesn't just store raw sensory data, it extracts meaning, creates abstract representations, builds concepts that exist independent of specific sensory inputs. Somewhere in your brain right now, there might be neurons dedicated to representing the people you love, the places you know, the ideas you think about frequently. Highly specific cells in vast networks, creating your mental model of the world through their selective firing. Your experience of recognition, seeing a face and knowing instantly who it is, emerges from these dedicated neurons responding to the presence of specific concepts. The basal ganglia, deep in your brain, learn habits through repetition until actions become automatic. When you first learn a skill, driving, playing an instrument, typing, it requires conscious attention. You must think about each action, deliberate over each step, but with repetition, something changes. The skill becomes automatic. effortless, almost unconscious. You can drive while holding a conversation, type while thinking about what you're writing, play music while focusing on emotional expression rather than finger placement. This transition from deliberate to automatic is mediated by the basal ganglia, structures deep in your brain that specialize in learning motor sequences and habits. As you repeat an action, the basal ganglia gradually take over control from the cortex, creating well-worn neural pathways that execute the action with minimal conscious input. This is wonderfully efficient, but also makes bad habits hard to break. Once the basal ganglia have learned a pattern, smoking after meals, biting nails when anxious, reaching for your phone when bored, that pattern becomes automatic, resistant to conscious will. The neural pathways are carved deep, executing automatically whenever the right trigger appears. Good habits and bad habits use the same mechanism. Right now, countless automatic actions you perform daily, the way you walk, how you hold a cup, your typing patterns, are controlled by your basal ganglia. You're freeing your conscious mind to focus on higher level concerns while automation handles the details. Your nervous system can adapt to missing senses. Blind people's visual cortex repurposes itself for enhanced hearing and touch. Your brain's regions have names that suggest fixed functions. Visual cortex for vision, auditory cortex for hearing. But these are more like default assignments than permanent destinies. If you're born blind or become blind early in life, then your visual cortex doesn't just sit idle. It repurposes itself, taking on new functions, processing information from your remaining senses. Blind individuals often show enhanced auditory abilities, better sound localization, better ability to discriminate subtle acoustic differences. Brain imaging reveals why. Their visual cortex activates during auditory tasks. The same for touch. Blind Braille readers show visual cortex activation when reading. Your brain regions are more flexible than their names suggest, adapting to whatever information they receive, making the best use of available cortical real estate. This plasticity reveals something profound. The brain's architecture isn't primarily about specific senses, but about processing patterns. Visual cortex is good at spatial information, temporal patterns, detailed discrimination. If visual input isn't available, it can apply those same computational abilities to auditory or tactile information. Your brain is opportunistic, adaptive, willing to redesign itself to make the best use of whatever input it receives. In the absence of sight, the visual cortex doesn't remain dark. It finds light elsewhere in sound and touch, and the rich non-visual world. The oldest evidence of a nervous system comes from fossils over 500 million years old. In the basic architecture of neurons, cells that generate electrical signals, that release chemical messengers, that connect into networks, is extraordinarily ancient. The oldest fossil evidence of nervous systems dates back over 500 million years to the Cambrian period, when complex animal life first flourished in Earth's oceans. Those ancient nervous systems were simple compared to yours. Nerve nets in early jellyfish-like creatures, simple ganglia in primitive worms. But the fundamental design was there, neurons communicating electrically and chemically, organizing into networks, processing information, controlling behavior. Evolution has been building on that basic design ever since, elaborating it, complexifying it, but never fundamentally replacing it. You're using neural technology that predates almost everything else about you. Your neurons work essentially the same way as neurons in ancient sea creatures from 500 million years ago. The same ion channels, the same neurotransmitters, the same action potentials. Your brain is built from ancient components, evolutionary solutions so effective they've remained essentially unchanged across vast spans of time. As you drift towards sleep tonight, your neurons operate through mechanisms older than bones, older than eyes, older than most structures we associate with animal life. Gap junctions allow neurons to communicate electrically without chemical neurotransmitters, while creating ultra-fast neural synchrony. Most neuronal communication involves the two-step process: electrical signal to chemical signal to electrical signal again. But there's a faster way. Gap junctions are direct electrical connections between neurons, channels that allow ions to flow directly from one cell to another without any chemical intermediary. Neurons connected by gap junctions can communicate almost instantaneously. This ultra-fast communication is used when precise synchronization is essential. In parts of the retina, gap junctions allow neurons to fire in perfect unison, creating coordinated responses to visual stimuli. In some motor circuits, they ensure muscles receive perfectly timed commands. In the brain, gap junctions help create the synchronized oscillations, brainwaves, that coordinate activity across large regions. It's a more primitive form of communication than chemical synapses, less flexible, much harder to modulate. But what it lacks in sophistication, it makes up for in speed and reliability. When neurons need to act as one, gap junctions make them literally electrically continuous, firing together with precision that chemical synapses can't match. Your nervous system uses both methods: slow chemical flexibility where that's useful, fast electrical connection where synchrony is critical, combining the strengths of each to create a communication network of remarkable versatility. Your brain's predictive coding system constantly generates expectations about what will happen next. You don't experience reality directly. You experience your brain's best guess about reality, constantly updated by sensory evidence. Your brain is running predictions. What should I see next? What should I hear? What should I feel? And comparing those predictions to actual sensory input. When they match, the world feels coherent and expected. When they don't match, you notice the discrepancy. Update your model, generate new predictions. This predictive coding happens continuously beneath consciousness. Your visual system predicts what the next frame of vision should look like based on the current one. Your motor system predicts the sensory consequences of your actions. Your auditory system predicts the next word in a sentence based on context. Most of the time, predictions are accurate, so you experience the world as smooth and comprehensible. But when predictions fail, things feel strange or surprising. This is why unexpected events capture attention. They violate predictions, forcing your brain to update its model. It's also why optical illusions work. They exploit situations where your brain's predictions differ from actual input, creating impossible experiences. Right now, your brain is predicting what will come next in this sentence, what you'll feel when you shift position, what you'll see when you blink. You don't notice these predictions unless they're violated. Your experience of the present moment is actually your brain's best guess about what's happening right now, continuously refined by sensory evidence. The locus coeruleus, a tiny cluster of neurons, produces most of your brain's norepinephrine and regulates attention. There's a small structure in your brainstem called the locus coeruleus, literally blue spot in Latin, named for its bluish appearance under certain staining. It contains only about 50,000 neurons, a vanishingly small number compared to your brain's 86 billion total. Yet this tiny cluster produces most of your brain's norepinephrine, a neurotransmitter crucial for attention, alertness, and stress response. Despite having so few neurons, the locus coeruleus projects throughout your entire brain. Each of its neurons can connect to hundreds of thousands of target cells, creating influence vastly disproportionate to its size. When the locus coeruleus activates, releasing norepinephrine broadly across your brain, your attention sharpens, your heart rate increases, you become more alert and ready to respond. It's remarkable that something so small, just 50,000 neurons in a brain of 86 billion, can so profoundly influence your entire state of consciousness. Activate the locus coeruleus and you're suddenly alert, focused, ready. Quiet it and attention drifts, alertness fades. Right now, as evening settles and sleep approaches, your locus coeruleus is likely reducing its activity, allowing the sharp focus of daytime to soften into the relaxed drifting of pre-sleep. This tiny blue spot in your brainstem, this handful of neurons, helps determine whether you're intensely focused or gently drowsy. Neural oscillations create brain waves that can synchronize across different regions during specific mental states. Your brain pulses with rhythms. When large groups of neurons fire together in coordinated patterns, they create oscillations, rhythmic waves of electrical activity that sweep across brain tissue. These aren't random fluctuations. They're organized rhythms with specific frequencies, and different frequencies correspond to different mental states. Delta waves, the slowest, dominate deep sleep. Theta waves appear during drowsiness and meditation. Alpha waves emerge during relaxed wakefulness. Beta waves characterize active, focused thinking. Gamma waves, the fastest, appear during intense concentration and may help bind different aspects of perception into unified experience. These rhythms can synchronize across distant brain regions, creating coordinated activity that links different areas into functional networks. It's like your brain is playing music, different instruments, brain regions, playing together in coordinated time. When you focus on a task, certain regions synchronize their oscillations. We're literally getting on the same wavelength. When you rest, the rhythms change, different regions finding different tempos. Right now, as you settle towards sleep, your brain's oscillations are slowing, shifting from the faster beta and gamma waves of active thought toward the slower alpha, theta, and eventually delta waves of rest. Your nervous system is a vast neurological orchestra, and consciousness emerges from its coordinated rhythms. The insular cortex processes interoception. your sense of your body's internal state, including hunger, thirst, and heartbeat. You know when you're hungry, when you're thirsty, when your heart is racing, when your bladder is full. But how do you know? These aren't things you can see or hear. They're internal sensations, your body reporting on its own state. This awareness of internal conditions is called interoception, and it's processed primarily in the insular cortex, a region tucked deep within the folds of your brain. The insula receives signals from throughout your body, your heart, lungs, digestive system, bladder, blood vessels. It integrates this information, creating your conscious awareness of your body's internal state. When you feel your heartbeat during exercise or anxiety, that's your insula at work. When you notice you're hungry or thirsty, your insula is interpreting signals from your body. Interestingly, the insula is also involved in emotional processing. Your feelings might be more connected to bodily states than you realize. Anxiety as awareness of increased heart rate, disgust as awareness of digestive responses, fear as awareness of physiological arousal. Your emotional life might be, in part, your brain's interpretation of what your body is doing. Right now, your insula is monitoring your internal state, your breathing rate, your heart rate, the fullness of your stomach, countless other variables, creating the background sense of how your body feels, the subtle internal landscape you usually only notice when something feels off. Axoplasmic transport moves materials along axons at speeds up to 400 millimeters per day, like a microscopic railroad system. A neuron's axon can be remarkably long, 3 feet or more in some cases, but the cell body, where proteins and other essential materials are manufactured, is at one end. How do these materials reach the far end of the axon, where they're needed to maintain the synapses and keep the neuron functioning? Through axoplasmic transport, a sophisticated cellular railroad system. Motor proteins literally walk along microtubules, internal structural fibers that run the length of the axon carrying cargo. Some motor proteins walk toward the axon terminal, delivering fresh materials. Others walk back toward the cell body, carrying waste and recycled components. And these molecular motors move at speeds up to 400 millimeters per day, slow by everyday standards, but remarkable for molecular machinery. This transport is essential. Without it, distant parts of long axons would wither and die, unable to maintain themselves. Disruptions to axoplasmic transport are implicated in various neurodegenerative diseases, where neurons slowly die because they can't maintain their far-flung processes. Right now, throughout your nervous system, the motor proteins are walking along axonal highways, carrying cargo back and forth, maintaining the infrastructure that allows neurons to function. It's a constant logistical operation, microscopic trains running endless routes to keep your nervous system supplied and functioning. The thalamus relays almost all sensory information to the cortex, serving as the brain's central switchboard. Nearly every sensation you experience, sight, sound, touch, and taste, passes through the thalamus before reaching your cortex. This oval structure deep in your brain acts as a central relay station, receiving sensory information from your body and sending it to appropriate cortical regions for processing. Visual information goes to visual cortex, Auditory information goes to auditory cortex. The thalamus sorts and directs traffic, ensuring information reaches the right destination. But there's one remarkable exception: smell. Your olfactory information uniquely bypasses the thalamus, going directly from your nose to the olfactory cortex. This direct connection might explain why smells trigger such immediate, powerful emotional responses and vivid memories. Scents access your cortex through a different route, one more directly connected to emotional and memory centers. The thalamus does more than just relay information. It also filters and modulates it, deciding what's important enough to reach consciousness. It's constantly evaluating incoming signals, amplifying some, suppressing others, shaping what you become aware of. Right now, your thalamus is processing sensory information from throughout your body, deciding what deserves cortical attention and what can be handled automatically. As you drift towards sleep, the thalamus changes its filtering, allowing less external information through, helping disconnect consciousness from sensory input so sleep can claim you. The consciousness may emerge from integrated information, the brain's unique ability to be unified yet highly differentiated. What creates consciousness? How does subjective experience emerge from electrical and chemical signals? No one knows for certain, but one influential theory suggests it has to do with integrated information. The way your brain is simultaneously unified and differentiated. Your brain is highly differentiated. Different regions do different things, process different types of information, yet it is also highly integrated. These different regions communicate extensively, combining their processing into unified experience. You don't experience vision, sound, touch, memory, and emotion as separate streams. You experience them as one integrated whole, one unified moment of consciousness. According to integrated information theory, consciousness emerges from this unique combination, a system that's complex enough to be highly differentiated, yet integrated enough that all the parts influence each other. The more integrated information a system generates, the more conscious it is. Your brain, with its 86 billion neurons forming trillions of connections, creating vast complexity while maintaining coherent unity, generates tremendous integrated information and thus consciousness. It's still a theory, not proven, but it offers a way to think about the mystery of subjective experience. Right now, your consciousness emerges from the integrated activity of countless neural circuits, working separately yet together, creating the unified experience of being you, reading these words, feeling whatever you're feeling, existing as one coherent point of awareness in the vastness of the universe. Your nervous system maintains a detailed body schema, which is why phantom limbs feel so real. Your brain maintains an internal model of your body, a body schema that represents where your limbs are, how they're positioned, how they can move. This schema is so fundamental, so deeply embedded in neural organization, that it persists even when actual body parts are missing. This is the basis of phantom limbs, the vivid sensation that an amputated limb is still present. People with phantom limbs don't just vaguely feel something is there. They experience detailed sensations, itching, pain, movement, position. They can feel their missing hand clench or their missing foot flex. The representation in the brain is so stable, so resistant to updating, that it continues generating sensations for a body part that no longer exists. This reveals something profound about how your nervous system constructs your sense of embodiment. Your experience of having a body isn't just passive sensation. It's an active model your brain maintains, a prediction of what your body should be like. This model is built early in development and can be remarkably resistant to change. Right now, your body schema is tracking the position of all your limbs, creating your sense of where you are in space, giving you the feeling of being embodied. This neural representation of your physical form is so fundamental that it persists even when the physical form changes. Neuropeptides can act as neurotransmitters, but persist in the synapse much longer, modulating neural activity for minutes or hours. Most neurotransmitters work quickly, released, bound to receptors, are recycled or degraded within milliseconds. They're like brief text messages, quick bursts of communication. But neuropeptides are different. These larger molecules can linger in the synapse for minutes or even hours, creating sustained modulation of neural activity. Neuropeptides don't just carry simple on/off signals. They modulate, adjusting the sensitivity of neurons, changing how responsive they are to other signals, altering the emotional or motivational context of neural processing. Endorphins, oxytocin, substance P. These are all neuropeptides that create sustained changes in how neural circuits function. This gives the nervous system two timescales of communication. Fast neurotransmitters like glutamate and GABA provide moment-to-moment signaling, rapid adjustments, immediate responses. Neuropeptides provide slower, sustained modulation, creating moods and states that persist, shaping the background conditions under which faster signaling occurs. It's like having both brief conversations and background music. The conversations convey specific information, but the music sets the emotional tone. Right now, various neuropeptides in your brain are creating the sustained background state, whether you feel relaxed or anxious, content or restless, against which your moment-to-moment thoughts occur. The cerebellum's parallel processing architecture inspired computer scientists designing artificial neural networks. When computer scientists began designing artificial neural networks, trying to create machines that could learn and process information like brains do, they looked to biology for inspiration. The cerebellum proved particularly influential. Its architecture, with millions of input fibers converging on relatively fewer output neurons, creating a system that can learn through repeated pattern exposure, became a template for early neural network designs. The cerebellum structure is remarkably regular and repetitive, easier to understand than the seeming chaos of the cortex. It processes information in parallel, handling many computations simultaneously, adjusting connection strengths based on experience, essentially learning from practice. This made it an ideal model for artificial systems. Modern AI owes a debt to your cerebellum, the neural networks that power image recognition, language processing, and game-playing AI use architectures inspired by biological neural systems, including the cerebellum's elegant design. Sometimes the best way to build intelligent machines is to copy designs that evolution spent millions of years perfecting. Your brain contains solutions to computational problems that human engineers are still trying to fully understand and replicate. As you rest tonight, your cerebellum continues its work, coordinating movement even in stillness, maintaining posture, using an architecture so elegant that it became a template for artificial intelligence. We still don't fully understand how anesthesia works, despite using it millions of times per day worldwide. Here we are, near the end of this journey through the nervous system, and we return to one of its deepest mysteries. Every day, across the world, anesthesiologists administer drugs that reliably, safely turn consciousness off. Patients go under, surgery is performed, consciousness returns. It works with remarkable consistency, and yet we still don't fully understand the mechanism. We know anesthetics disrupt neural communication, that they interfere with certain neurotransmitter systems, that they may prevent the brain regions from synchronizing in ways necessary for consciousness. But there's no complete unified theory that explains how these drugs achieve what they do. Different anesthetics seem to work through different mechanisms, yet all accomplish the same goal, the temporary elimination of consciousness. It's humbling. One of medicine's most essential tools, used millions of times daily, trusted with people's lives, remains partially mysterious. We can reliably control consciousness without fully understanding what consciousness is or exactly how we're controlling it. The nervous system keeps its secrets, even as we learn to manipulate it with increasing precision. Perhaps this is fitting, that consciousness, your most intimate and immediate experience, remains somewhat mysterious even to those who can temporarily take it away and return it. Every thought you've ever had, every memory you cherish, every person you've loved exists as specific patterns of neural firing. And so we arrive at the final fact, the one that encompasses all the others. Everything you are, every thought you've thought, every memory you hold, every person you've loved, every dream you've dreamed, every fear you've felt, every moment of joy or sorrow or wonder exist as patterns of neural firing in your brain. You are, in the most literal and profound sense, made of electricity and chemistry. Patterns of ions flowing across membranes, neurotransmitters crossing synaptic gaps, waves of electrical activity sweeping across neural tissue. These patterns are you. Your personality is a pattern. Your memories are patterns. Your sense of self is a pattern, remarkably stable yet constantly shifting, maintained by the continuous activity of your nervous system. This could feel reductive, diminishing, reducing the richness of human experience to mere biology. But perhaps it's the opposite. Perhaps it's astonishing that such patterns can create the vast internal universe you inhabit, the profound depth of human experience, consciousness, love, creativity, wonder, all of it emerging from patterns of neural activity, from the coordinated dance of 86 billion neurons, made from the ancient mechanisms of electrical and chemical signaling refined over 500 million years. Right now, as you read these final words, you are patterns firing, experiences emerging, consciousness persisting. You are your nervous system, and your nervous system is you, fleeting, precious patterns dancing through biological circuits, creating the miracle of awareness, of being someone at all, of experiencing this strange and beautiful universe from the inside. These patterns will continue as you drift towards sleep, shifting into the slower rhythms of rest, but never entirely stopping, never completely silent, maintaining the continuity of you through the darkness, through the dreams, ready to wake and pattern again when morning comes. And so our journey through the nervous system comes to rest. We've travelled from the vast 86 billion neurons, networks more complex than galaxies, to the impossibly small synaptic gaps measured in nanometers, near single photons detected by retinal cells. We've explored ancient mechanisms that predate humanity by 500 million years, and cutting-edge discoveries made just decades ago, overturning what we thought we knew. Throughout it all, there's a thread worth holding. The nervous system is both entirely physical, ions and molecules, electrical gradients and chemical messengers, and somehow transcendent, creating from those physical processes the entire interior world of human experience. You are patterns in motion, chemistry and electricity organized into something that can wonder about itself, that can lie here in the quiet of evening and contemplate its own existence. Perhaps what's most remarkable is that your nervous system does all this work, maintaining your heartbeat, processing sensations, storing memories, generating thoughts, without asking for recognition or rest. It's been with you since before your first breath, working faithfully through every moment of your life, creating the continuous experience of being you. As you settle into sleep now, your nervous system will shift its rhythms but never fully stop. It will consolidate today's memories, clear metabolic debris, coordinate the slow rhythms of rest. It will maintain your breathing, your heartbeat, the essential functions that keep you alive, all while you drift through dreams, consciousness temporarily released from the waking world. Sleep well. Your 86 billion neurons have earned their rest, and they'll be ready, faithfully, inevitably, to create another day of consciousness when morning comes. Thank you for spending this time exploring the extraordinary system that makes you possible. Until next time, may your thoughts be gentle and your sleep be deep. Good night.